Okay, so Assalamu alaikum everyone. My name is Jana Atef. I'm a third year medical student, and today we are going to be talking about the histology of the muscles, the lymphatics, and the nerves. Okay, so histology, I know unfortunately that a lot of you don't like it or are like annoyed by it, but honestly, it's like easy marks, you just need to know a few keywords, a few stuff that you need to know, some pictures, and you're good to go, honestly. Okay. So this PAL is basically like a revision for you for your final. I know your final is coming up in a couple of days, not to scare anyone, but I made this so it's like a revision for everything, including these three lectures. So that just like a night before the exam or whenever you want, you can go over it, you can watch it, the recording, whatever it is, and you're ready to just go in with the histology for these specific lectures. Okay. So Bismillah ar-Rahman ar-Rahim, let's start. Okay, so we're gonna start first with muscles, okay? So muscle histology is very easy. It's just a few concepts that you guys already took in physiology, okay? And yeah, very muscular. So we're gonna talk first about the basics of the muscles, okay? Like I said, you guys took most of it in physiology, everything about like actin, myosin, these kinds of stuff, which is a huge bulk of the lecture. It's actually like the most difficult part of this lecture. So if you already know that from physiology, this is gonna be very easy. Okay, so for muscles or the muscle cells, what we need to know is that muscles are contractile cells, okay? Why are they contractile cells? Is because of the actin and the myosin. Again, if you remember from physiology, the actin and myosin filaments, all these physiology of the muscle contraction, this is what makes them contractile, okay? The embryological origin for muscles, this was something that was mentioned in your slide at some point. It's mesoderm. So if you ever got like a random MCQ about that, what's the embryological origin of muscle cells? The answer is mesoderm. Okay, they're mesoderm in origin. Now, there's some different namings for muscles. Okay, you know how we have in our histology or the cells or everything, there's a cytoplasm, there's an endoplasmic reticulum, there's a cell membrane. So for the muscles, they like to be a little bit special. This is also the same with nerves when we go to them, but for muscles right now, they have different namings, okay? So the cytoplasm for the muscles is called the sarcoplasm. For the endoplasmic reticulum, it's called the sarcoplasmic reticulum. And for the cell membrane, it's called the sarcolemma, okay? It's just you just need to know them. It's not that hard. Just know that when I say sarcoplasm, it means I'm talking about the cytoplasm of a muscle cell. I'm talking about the sarcoplasmic reticulum. I'm talking about the endoplasmic reticulum of a muscle cell. And for the sarcolemma, it's basically the cell membrane of a muscle cell. Okay. Now, the main part of this, Yani, is the types of muscle cells. Okay, so we have two types. We have striated and non-striated muscles. Okay. Now, the striated, does anyone know what striated means? If you know, just write in the chat or raise your hand or anything. Okay, anyways, the striated muscles, it basically means it has stripes, okay? So the muscles that have stripes, or the ones that are striated, are the skeletal and the cardiac muscle cells. Okay, we're going to talk about why in a bit, but just know that the muscle cells, when we're talking about that are striated, they're the skeletal and the cardiac, okay? If they're non-striated, we're talking about the smooth muscle. So anytime we mention non-striated muscle cells, we're talking about smooth muscle cell, okay? That's just a picture of the arrangement. This is a skeletal muscle, and we're talking about why it is that way. So, like I said, let's start with what I was saying, the muscle cell, the skeletal muscle cells, okay? We said they're striated. This is exactly what striated means. Look at these like lines. They're just a lot of lines, okay? That's what striated means. It reminds me honestly of like a snake tail or something. So whenever you see any striated muscle, it could either be a cardiac muscle cell or a skeletal muscle cell. How you differentiate them? Okay, so first for skeletal muscle, how do you know this is a skeletal muscle? One, it's striated, like we said. Two, it's multinucleated. So this entire thing is a muscle cell, okay? And if you get like a section of your, for example, your any muscle in your body, your sternocleidomastoid muscle, you take a section of that and you put it under the histology, the entire length of the muscle is one muscle cell, okay? It's a muscle fiber or muscle cell. So the entire thing, that's one muscle. So obviously you can't have just one nucleus in it. It's not going to be enough for it to have the energy. So it's multinucleated because the muscle cell is just like so long, it needs multiple nuclei. So whenever you see multinucleated cells, which is like more than 3 nuclei, it's probably a skeletal muscle, okay? Other thing is that the nucleus are found pushed close to the sarcolemma. And what did we say the sarcolemma was? It was a cell membrane. So here, this is like a schematic diagram of it. The nuclei are all like on the edge, It's right here. It's called a peripheral nuclei or peripherally pushed or anything that you want to call it. But it's basically when you see a cross section, this is a cross section of a skeletal muscle. You see these like dots. These are the nuclei. So you see them. They're all pushed in periphery. There's nothing in the middle. Okay. This is the opposite for cardiac muscle, which we're going to talk about. These ones they have their nuclei in the middle. But these ones, the skeletal muscles, the nucleus is close to the periphery. Okay, so again, cross section of it. The nuclei are all in the periphery. This is a very common OSCE question, by the way, OSCE question. It could get you just a cross section. Whenever you see peripherally pushed nuclei, you see striated cells, you see multinucleated cells, it's most likely a skeletal muscle. Okay. Next is the levels of organization. So for me, honestly, this was a very hard concept to grasp because there's just a lot of names and a lot of small inside. So you can think about them like the Russian nesting dolls, okay? The big doll is the muscle. So here, this is the big one. This is a muscle, the entire muscle. Okay, inside it, it's divided into fascicles. So these groups inside the muscle cell, the muscle itself, like the big muscle, these are called the fascicles, okay? And what is inside the fascicles? You have the muscle fibers, okay? And the muscle fibers are the muscle cells, okay? So inside, so this is a fascicle, okay? Inside them, these little dots inside the fascicles, these are the muscle fibers, okay? Now inside the fibers, there are the myofibrils, okay? You think you can see them here. So this is a muscle fiber, okay? This is like the entire muscle. See, here's the nuclei. And yeah, this is the muscle cell itself. Now, inside it has the myofibrils, okay? Myofibrils are the contractile units, okay? They're the ones that have the sarcomeres, which you guys also should have studied in physiology. The sarcomeres are what causes it to be contracting. So inside the sarcomeres, we have the myofilaments, okay? So don't confuse myofibrils with myofilaments. Myofibrils are inside, like they're directly inside the muscle cell. So this is the muscle cell. Inside them are the myofibrils, okay? Now inside the myofibrils is the myofilaments, okay? You can think about it like fibrils, it's a small name, but it's bigger than the filaments. It's like opposite kind of thing. Myofilaments, it should be the big one, but it's actually the small one. The myofibrils are the small ones. The name is small, but they're actually the bigger ones, okay? In terms of like the hierarchy. So again, big one is a muscle cell. Inside it, you have fascicles. Fascicles are the groupings of muscle fibers, okay? So the groups of muscle fibers are a fascicle. Now, inside the muscle fiber is a myofibril. The myofibrils are the sarcomeres that contain the sarcomeres, okay? Inside the sarcomeres, you have the myofilaments, okay? Myofilaments are the thick filament and thin filament, okay? This is from the sliding filament theory. These are the actual contractile units, okay? So the thick filament is made of myosin, and the thin filament is made of actin. Now, these names are much easier to remember. Actin is like tiny, okay? It has a T-I-N. Think of it as a thin, okay? The thick filament is myosin. It's like a bigger name, myosin. It's the big one. So thick filament is myosin, thin filament is actin, okay? Now, something else that we didn't mention is the coverings of them, okay? This is another picture. So the myofibril are the tiny ones. Muscle fiber, that's inside a muscle fascicle, that's inside a muscle, okay? Now the coverings of them. So the first covering you have, here are, okay, the connective tissue. You've taken the connective tissue in the lecture before that, I think the PAL before this one. So you should know what they are. So there's epimysium, perimysium, and there's endomysium, okay? From their names, epi means that it's the big one. It's the epimysium that surrounds the entire muscle. So this entire skeletal muscle, what's wrapping around it is the epimysium, okay? So this entire white, under the deep fascia is the epimysium. That's wrapping around the entire muscle, okay? It's made of dense, irregular connective tissue, okay? And it extends into the septa. So if there's like some septa separating some big muscles from them, from each other, that's epimysium, okay? Now the perimysium, it's what separates the fascicles, okay? So where is it? This is right here. We said this is a fascicle, okay? If it's inside the big muscle, these groupings, these are the fascicles. So what wraps around the fascicles? The perimysium, okay? Peri means in the middle. So the epi is a big one, okay? Peri means middle, so it's not the small one and it's not the big one. It's the one that wraps around the fascicles. And the last one is the endomysium. The endomysium is the one that wraps around the muscle fibers, okay? So each fiber is wrapped by endomysium, okay? Right here, this is a histology slide of it. So this is a connective tissue. This is a myofibers, okay? Myofibers are muscle cells. So when you see they have a nuclei, multinucleated, okay, this is a muscle cell. Muscle cell is also a muscle fiber. So what wraps around each of them is the endomysium. What wraps around the fascicle that contains him, that's the perimysium. What wraps around the entire muscle is the epimysium. Okay, it's very simple once you get like the names. You have epi, peri, and then endo. Okay. Now, question. So what is the cause of the striations of a skeletal muscle cell? Remember, we said we're going to discuss more of it. We didn't mention what they are, but this is like an introduction to what we're going to discuss next. What are striations made of, or why is it striated? Okay. Does anyone have an answer? Let me check the chat. Actin and myosin. Okay, that's close. Sarcomeres. Okay, yes, sarcomeres. But what part of the sarcomeres exactly? Okay, so the answer is they're due to the dark A bands and I bands. Okay, so yes, it is a sarcomere. Sarcomeres, we said, like the structure of it is made of alternating A and I bands. Okay, and between them is like the Z lines. So this is the reason why it's striated. So right here, this is a clear picture of it. See the striations? Okay, you zoom into them. This is a light, dark, light, dark, light, dark. Okay, what is that? This is the A and the I. Okay, so the A is a dark one. The A band is a dark. We said dark means thick filaments or myosin. Okay, and the I is a thin one that contains a thin filament, which is the actin, and it's also a lighter band. Okay, so what was a sarcomere? Does anyone remember what a sarcomere is? It's right here in the slides too. Okay, sarcomeres were from one Z line to the other Z line. What's a Z line? The Z line is in the center of an I band. Okay, so let's like backtrack a little bit. This entire thing is a piece of the striations. Okay, so this one is a sarcomere. How do I know this is a sarcomere? Because I have two Z lines. So between two consecutive Z lines, this is a sarcomere. This is the contractile unit of a muscle. Okay, again, remember from physiology, sarcomere is what causes a muscle to contract. So sarcomere is the area between two Z bands, okay? Now, the A band and I band, we said this is the reason for the striations because the A band is dark and the I band is light, okay? Now, what makes them light and dark? We said that the I because of the actin and the A because of the myosin, okay? Now, there's some zones inside there, just like in the middle. The H zone, basically, it's the lighter region inside the A band. So we said the A band is dark. Yes, it's dark. But in the center, if there's a light part of it because there's no overlap. Okay, I think I have a better picture. Yeah, right here, Z line to Z line. So this is inside it. This is a sarcomere. Okay, A band we said it's from it's a darker one. So the one that has overlap of actin and myosin. This area right here, you guys see it? The thin one is actin, the thick one is myosin. Where there is overlap, it's dark. Where there's no overlap, this is the M line or the H zone. Okay, the middle of it exactly is M line. Around it, the area where there's only myosin, this is called as the H zone. Okay, so again, why is it H zone? It's because it's the area that is overlapping of actin and there's a question, would the HM become darker during contractions? Yes, it would. Why? Because we said while they're contracting, there's gonna be more overlap. So where there is overlap, it's gonna be darker. Okay. So again, the H zone, it's lighter in the middle of the dark one because it has no thin filaments. Okay, there's no actin, it's only myosin. Now the I band, let's go back to the I band right here. I band actually has some, it doesn't have any. Okay, so that when we say where is the area that contains only myosin, that's the H. So, where is the area that has only actin? That's the I band. Okay, what's the difference between the M line and H zone? Good question. Basically, it's not different in that they're like different structures. They're related to each other. So basically, M line, let's see on the other side. M line is just a line of proteins. Okay, it's the center of it exactly. So if you take a sarcomere and you fold it in the middle, this middle is called an M line. It's just a way for pathologists or any histopathologist, like doctors, to understand like where each thing is. It's just like the middle of the A band. Okay, the H zone, like we said, H zone is only myosin. So where there is no overlapping from actin and myosin, that's called the H band. H band is found where? It's inside the A band. Okay, so basically, you take the A band, it's all of the myosin. You go in the middle just until you stop finding any actin, this is the H zone. Do you get it? I hope that's clear. Okay. All right, perfect. So basically, this is the arrangement of the sarcomere. Okay, if you guys don't understand it, let me know. I can repeat it, but it's very easy. The striations are caused by what? The alternating A and I bands. Okay, H zone, M zone, we discussed that. So let's move on to the next. Now the cardiac muscles, okay? So we finished the skeletal muscles. The skeletal muscles, we said they are striated, they're contracting. And one more thing I'd add to the contracting part of it is that it's voluntary. This voluntary means you can control it, okay? Now the cardiac muscle cells, they're also striated. Like we said, what's the one that's non-striated? Pop quiz. Can you explain what's a Z-line? Sure. Z-line is basically, it's called also a Z-disc. It's what tells you this is a sarcomere. It's basically like the end of a sarcomere. So from one Z line to the other Z line, this is sarcomere. Think about it, it's like an idea, okay? It's also some proteins. So like the M line, it's also proteins. It's what like attaches two sarcomeres to each other. So between two sarcomeres, there's a Z line. Two sarcomere, Z line. So it's like they don't hold hands together. They don't like that. So they have to wear some gloves, you know? So when they're holding hands together, each sarcomere to make the entire muscle cell, this is a Z line. These are the gloves, okay? So again, between two Z lines is a sarcomere. Did you get it? Perfect, okay. Again, so muscle cells, it's striated, okay? And remember we said skeletal muscle were multinucleated? These ones are not. They usually have only one nuclei or sometimes two, but not more than that, okay? They also have a branching pattern, okay? You guys need to look at this picture very well because it came to us in the OSPE, okay? It was a picture of this one, and beside it, I think was a skeletal muscle. So the only difference that you can tell, because this one was branching, okay? Let's go back to the skeletal muscle. Where is it? Yeah, skeletal muscle is just like one line, just like a tube. It doesn't branch, doesn't do anything. It's continuous line. Like I said, if you take a piece of your muscle, any muscle, and you do it histologically, the entire muscle fiber or the muscle cell runs along it. So it's just one long cell. Like it's a really long cell. Now the cardiac muscle, it branches. Why? Because like you think about it as like the heart is just one it is one muscle. It's a very special muscle. It's called myocytes, okay? So the heart muscle cells are called the myocytes, okay? And when you take like the physiology of it in CVP, inshallah, you're gonna know that there are the myocytes they contract by themselves, not automatic. I mean, like, okay, myocytes, they don't need nerve junctions, okay? So that's why they have something called intercalated discs, okay? So if you have a question, why do muscle cells contain intercalated disc? It's so that they can pass the action potential between each of them. If you recall from physiology, the action potential goes from when there is a neuron and it attaches to the muscle cell, okay? And this junction, like there's the passing from the transmissions or the neurotransmitter, whatever it is, and it causes action potential across the cell, okay? For the skeletal muscles, each cell has its own like synapses with the axons of a nerve. For cardiac muscle, no. Cardiac muscles, they take the original action potential from something called the pacemaker cells. They're the ones that start the action potential, okay? And then it like kicks the cell right beside it, and this one causes like an entire loop. It's like a domino effect, okay? So cardiac muscles, they're not innervated like the rest of them. They go by the intercalated discs. So intercalated discs are like the bridges, okay? So like I said, so take this piece, okay? Look at this one. This is one cardiac muscle cell, okay? If this one got depolarized from the one adjacent to it, it's gonna depolarize the one after it. How? Because of the intercalated discs, okay? Skeletal muscles don't have that. They each have their own neuron attached to it, okay? Each muscle is innervated, okay? This one, no. All of them is second a dominant effect, like I said. So how does that happen, or what gives them these property? The intercalated discs, okay? Intercalated disc, so in histology, it just looks like lines, basically. So we said they're striated, so don't let it confuse between the striations and the intercalated discs. If you see here, these are like very thin lines. This is the striations. These are the A band and I band. Now the thick line in the middle between like the muscle cells, this is the intercalated disc, okay? Again, so this is the electron microscope. This entire like zigzag, that's an intercalated disc, okay? This small A band, I band, this is very clear, like the sarcomeres, because this is two Z lines, between them is a sarcomere. These are the striations, okay? The one between them, like big bridge, that's intercalated disc. All clear? This picture came to us in OSP, this exact picture, and this one. This one's like the branching, okay? So look at it, it branches. It's not one continuous muscle. It like goes on and it branches, okay? Intercalated disc between them, it's also very clear. So make sure if you see this in an OSP, you know it's a cardiac muscle. And if it tells you why do you think this is a cardiac muscle cell, you say 1, they're intercalated discs, 2, it's branching, 3, it has only 1 or 2 nuclei. You see here, this is one muscle has its own nuclei. This muscle has its own nuclei. This one has its own. You get it? Okay. So would it be similar to maybe a road of Ranvier in the sense that it allows synapses to move through? Similar concept. Yes, I'd say it is a similar concept to like the nerve cells, like the jumping through them, but it's not the same mechanism. Like it doesn't actually jump. This is more chemical. In the nerve one, it's more electric, okay? This one's more chemical. It's just like think about it as a gate, okay? So like I want to move from this room to this room, and there's a door in between. This door is integrated desk. So if I want to move through, me being the actual potential, I'm gonna open the door, I'm gonna pass through. You get it? So A and I bands are found in all muscles. They're found in all striated muscles. Again, why are they striated? Because they have A and I bands, okay? If they don't have A and I bands, means they're not striated. And what are not striated? Smooth muscle cells, okay? Smooth muscle cells, we're gonna go to them like right after this one. They're not striated, which means that they don't have striations or A and I bands, okay? What are the intercalated discs made of? They're made of junctional complexes, okay? I think it was another lecture that you have that. There's a different types like desmosomes and junctional complexes. This is what they're made of, okay? Like I said, think about it as a door. These kinds of junctions and desmosomes, they're the doors. So that's why they're so big, because it's their own structures. Okay, you got it? Okay, so let's move on. This is all good for cardiac muscle. Like I said, you see this, guys, it's so easy, 3 points. This is cardiac muscle, okay? Perfect. Now the smooth muscles, okay? So like someone asked, all muscles have A and I bands? No, the ones that don't are these ones, okay? The smooth muscle cells. Smooth muscle cells are unstriated, means that they don't have any striations, which means that they don't have A and I bands, which also means that they don't have sarcomeres. Why? Because the sarcomeres and the A bands, the sarcomeres, all this arrangement is mainly for the cells that have like very specific type of contraction. Smooth muscle cells, they're involved with like the um autonomous, yeah, the nervous system, which basically means it's involuntary. You can't control it, okay? So where are they found? This is gonna make more sense. Blood vessels, respiratory tract, GIT, and the urinary system. Blood vessels like the arteries and the veins. Can you control your own arteries? No. If you can, that's a big problem. Respiratory, like the bronchioles, sometimes the alveoli, these structures are made of smooth muscle cells. GIT, the prime example of smooth muscle cell is the small intestine, okay? If you touch it, if you see us, I think you guys saw it in lab, the smooth muscle cells of the intestine, it's just smooth. Like that's how it looks like. It's a smooth muscle, okay? It has no bumps or lumps or there's no nothing, just like one smooth sheet of muscle, okay? This is the exact opposite of skeletal muscle. Skeletal muscle, if you touch them, they're rough, okay? They're dense. They have some core to it, you know? So the cells themselves, here's a picture of the cell. This is a smooth muscle cell, okay? They're fusiform. What does fusiform means? It means that it has a thick center and tapering edges, okay? So this is exactly how it looks like. It's a smooth muscle cell. One cell that looks like that, okay? Histologically, right here. This is a cross section of it, okay? Again, this is a cross section. You just I put these pictures for you so you guys can familiarize them. So if you see them in an OSP, you know what this is. So this one, look at it. It's like a central nucleus. So how would you tell the difference between this one and skeletal muscle cross section? The nucleus. Okay, remember when the skeletal muscle we said it's pushed to the side? This one is right in the middle. It's like smack in the middle. Again, this one is also clear. It's just different kind of stain. And right here is the transverse cut of it. Here, there's just like long muscles. Each one has its own nucleus, and yeah. Back to it, it's under the control of the nervous system. Okay, so it has no neuromuscular junction. It has no sarcomeres. It has its own mind and own everything. Okay, can you see the striations? Every type of cut. No. So for the cardiac and the skeletal, you can see the striations if it's a longitudinal cut. Okay, so like the muscle cell, it's like this, not a cross section. Cross section, you wouldn't see the striation because you think about it, striations are on the outside. Okay, so if you look at it from the inside, you're only going to see the nucleus and the inside of it. So this is a good question. So if you get a cross section of a muscle, okay, how could you tell that this is a skeletal muscle or a smooth muscle or a cardiac muscle? Again, let's look at the pictures so we can better explain this. I don't have a picture of a cardiac muscle in cross section, but they usually don't get it as cross section. You're only going to see it like that because the main identifying features of cardiac muscles are the integrated discs and the branching pattern. Okay, skeletal muscle, where's the cross section? Right here. Look at the nucleus. It's pushed to the side. You see a nucleus is pushed to the side. That's a skeletal muscle. You see a nucleus that's in the middle. Where is it? Here, this is a smooth muscle. You get it? Let me see a chart. Yeah. So if the nucleus is in the middle, it's a smooth muscle. If the nucleus is pushed to the side, it's a skeletal muscle. Okay. So these ones are lined by gap junctions, okay, which permit synchronous contraction of them. What does that mean? It means like similar to the cardiac, but it's not cardiac, okay? Because they're controlled by like the hormones, they're controlled by their own other systems. So this is not very high yield, but just know that the smooth muscle cells or smooth muscle organs, they have their own mind, okay? They don't follow the voluntary actions or like the skeletal muscles or the cardiac muscles, their own different family, okay? So the ones in the blood vessels, this is important information. The smooth muscle cells, we said they're in the blood vessels, okay? These are the arteries and the veins. They synthesize collagen, elastin, proteoglycans, and multi-adhesive glycoproteins. Basically, what that means, it has fibroblastic activity or clotting activity, okay? You're gonna take that inshallah in CVP again. There's like endothelial damage, it's gonna cause the coagulation cascade, all of that. But just know that they have special identities, okay? So smooth muscles, no, it's unstriated. The second you see unstriated and the central nucleus, that's a smooth muscle. So identifying is easy, okay? Where is it? Blood vessels, respiratory, GIT, urinary system, and maybe and the uterus too, okay? And they're controlled by hormones. So an example of that is the uterine contractions, okay? So during labor, for example, the brain releases oxytocin. This is a hormone that allows the woman to like have contraction. So once oxytocin goes to the smooth muscle cell, it's going to cause them to contract, okay? So you see how they're not controlled by the neurons or anything, it's controlled by hormones. So this is how like they're different. And different organs have different hormones. Okay, so for example, in blood vessels, there is a hormone called the nitric oxide. It causes the constriction or the dilation. I forgot, but like basically they're controlled by their own systems. Okay, again, let me see if there is a question. Are cardiac muscles involuntary? Yes, they are involuntary too. Both of them are involuntary. What's it called? Cardiac and smooth muscles, but a different kind of control. Like I said, smooth muscle mainly like hormones. It's more like its own system. So does the cardiac muscle. I'd like to think of the cardiac muscle or the heart itself. It's like an entire different system. Okay, so yes, it's involuntary. You can't control your heart. Again, these are the pictures. Familiarize yourself with them. And this is a comparison slide. Okay, so this is very, very useful. So skeletal muscles, what do you see here? Okay, I want someone to point out one feature or two features that you can tell that this is a skeletal muscle. Yeah, any feature, whether from the cross section or from the transverse section. Multinucleated, that's right. Yes, look, this is entire muscle. Striations, yes. Nucleus on the periphery. Exactly, you guys said all of them. So in the cross section, we have the nucleus on the periphery. On the transverse section, there are striations, okay? And one more thing is that it's not branching. You see how this is just a continuous cell? Parallel non-branching, exactly. Yeah. This is, mashallah alaikum, guys. This is the skeletal muscle, okay? Cardiac muscle, okay, same thing. I want three features for cardiac muscles. Integrated discs, branching, yes. IDs, yes. Exactly, exactly. Yes, 1 to 2 nuclei. That's it. So we look at it, it's branching. You see this one is moving into the next one, and this one's moving, it's like all of them are connected to each other and has integrated discs, okay? What was the function of integrated discs? Why is it there? What aspect of a heart needs it? Passing of, yeah, exactly, okay? Acid gas junctions, exactly, yes. Mashallah, guys, you're doing really well. That's exactly how it is. So action potential, when it starts from the pacemakers, for it to pass through the cells, it passes through the integrated discs, okay? Now, last one, smooth muscle. Give me two aspects of it, okay? Fusiform, exactly, yes. What else? No striations, yes. One nucleus, okay. Non-striated, yes, exactly, you guys got it. And a central nucleus, exactly. That's it. That's basically it, okay? So mashallah, you guys got everything for the muscles, okay? You know how to identify them. So if it came to you in an OSPE, will you be able to identify them? Yes, no? Yes, inshallah, inshallah. It's very easy, okay, guys? Like I said, it's 3 points. Make sure just like look at the pictures multiple times. If you ever forget it, think about the physiology of it, okay? The heart, the skeletal muscles, smooth muscles. When you think about like the function and the structure, it really makes sense, okay? Okay, so we're done with the muscles, okay? Now we're gonna move on to the nerves. Okay, nerves are like the fancy ones. That's how I like to think about them. Okay, so some info that you need to know is that the cells of the nervous systems, okay, there are two kinds of cells, or there are two families of cells we find in our entire nervous system. You guys took already a lecture about the nervous system. So there's the CNS and the PNS, right? The central and the peripheral, okay? Can someone tell me what was the central nervous system? What is the central nervous system? Brain and spinal cord. Yes, exactly. And the peripheral nervous system was the peripheral nerves, okay? So the entire systems of the nervous system have two kinds of cells. They have the neurons, okay, or the nerve cells, and they have the glial cells, okay? And this is basically what this entire lecture is for. There are glial cells and there are neurons. Neurons are the main neurons, okay? These are the ones that you know, the ones that we're all familiar with, the axons, dendrites, soma. These are the neurons. The neuroglial cells or the glial cells, we're going to go more into it, but it's basically the supporting cells, okay? So this is a neuron. This is a glial cell. So first of all, neuron. It's easy, okay? You guys probably know this. So could we by any chance ask? Yes, you can. We'll send you the slides, inshallah. This is a neuron. Okay, so what are the parts of a neuron? Okay, it has a cell body, it has dendrites, and it has axons. Okay, I'm pretty sure you guys are gonna know that you know all these information. So let me ask the question: what's a cell body, or what is it also called? The cell body of a neuron. What is it called? Soma. Yes, a soma. A soma is a cell body of a neuron. Okay, what does it contain? Where are the slides going to be sent? The slides are going to be sent, inshallah, on email, if I'm not mistaken. I'll let you guys know, and yeah, I'll let you know. It contains a nucleus, yes. So the cell body of the neuron, it has a very large nucleus. It's something very distinct of neurons. It has a very large nucleus, okay? You look at it right here. I think I have another picture. Yes. Where is it? This is a nucleus. Look at it. It's huge. It's taking up almost half of the cell, okay? And it also has a really large and prominent nucleolus, okay? These parts, like, they're found in all cells, okay? All cells have a nucleus and has a nucleolus, except red blood cells. But I mean, like, all cells of your body, they have these kinds. But a neuron has a very big, okay? So if you have a question that's like identifying a neuron, or like what's an identifying feature of a neuron, you can mention that it has a really large nucleus. That's considered correct, okay? The other thing is that it has some basophilic areas. What does basophilic means? It means like dark blue areas right here. They're called nestled bodies. What are nestled bodies? They're like areas or aggregates of rough endoplasmic reticulums and polyribosomes. Okay, yes, exactly, and ribosomes. So you have any clumps of these RERs and the polyribosomes, or just ribosomes. These are called nestled bodies, and they're found where? They're not found in the axons, they're not found in the dendrites, they're found where? In the cytoplasm of the soma. Exactly. It's found in the cytoplasm of the soma. This is where you find the nestled bodies, okay? Here it is, another identifying feature of a neuron. Okay, why do I keep mentioning identifying features? It's because a very common question in OSPE is that they're going to give you a picture. So for example, if they gave you this picture, they're going to be like, it's going to come in two parts, of question. It's going to be like, what is the cell? Or like, identify the cell. And the second question, it's going to be like, mention two identifying features, or 2 to 3 identifying features of the cell. So like we said in the muscles, how we have each one, they have their own identifying features. The neuron also has. Okay, one of it is that a very large nucleus, a prominent nucleolus, and also has nestled bodies. Okay. Now the dendrites. Okay, dendrites. Okay, question: do they receive or do they send information? There's a spoiler in the slide, but just try to think about it. Both. Okay, that's interesting. Receive. Okay, mainly we're gonna stick with receive. I'll tell you when does it do both. Like, I get where you guys are coming from, but dendrites are the ones that receive stimuli. Okay, if they are the dendrites, the actual protrusions from the cell body, they receive stimuli from other neurons. Okay, now, yeah, this is this is from the nestled body specific to specific kinds of. Yeah, there aren't very obvious in the pictures, honestly, but no, they should be found in all kinds of neurons. This is basically the basic structure of a neuron. So everything mentioned here is found in all neurons, okay? But some of them, yeah, you're right, they're not, because they need a special stain. It doesn't show in all stains. See this one, how it's blue? I forgot the name of the stain, but it's clear only in certain stains, okay? Now the axon. Axon is a long part of the the neuron, okay? And what does it do? The entire axon body, it's the one that conducts the signals, okay? The action potential. Where does it move in? It moves in the axon, okay? So you see this thing that I wrote here, only one in each neuron. Some of you are going to be confused because there's something called pseudo, where is it? Here, pseudo unipolar. It's still one. All neurons have one axon. Okay, so this one, let's look at a multipolar. Multipolar means here, there's one axon. Bipolar, there is one axon. This is a dendrite. This is not an axon, okay? So bipolar means it's the body, the cell body, the soma separates the dendrite from an axon. It just has a very long dendrite, okay? Now the pseudo-unipolar, it's one long axon, and just like the cell body, it's like, it's elongated from the side, okay? It's just from one side, it has cell body. This is honestly a weird kind of neuron. I don't think it's even in humans, but yeah, that's it. So all neurons have one axon. And it starts with something called the axon hillock. Okay, this is always a question, guys. The question was, where does an action potential start, or the propagation of an action potential? Where does it start? It starts in the axon hillock. Okay. One identifying feature of an axon hillock is that it has no rough endoplasmic reticulum. Where did we say they were? Where did we say we have rough endoplasmic reticulum and ribosomes? What was the structure called? The structure we in the soma, yeah, but why are they called like the clumps of them? Nestle bodies, exactly. So in the soma, you have nestle bodies. Now, once we start with the axon, which is the first part, which is the axon hillock, you stop having rough endoplasmic reticulum. Okay, it's just an aspect of it. It stops having rough endoplasmic reticulum, and this is the start of the axon hillock. Okay, start of an action potential is at the axon hillock. Okay, now we're good with this one. These are just pictures. Again, there's the nestle bodies. Dendrites, they're extensions from the soma itself. And the nucleus, like we said, it's a big nucleus with a prominent nucleolus. We already discussed this one. They all have one axon. It's just like where the soma is located. And then the synapse. Okay. Now synapse, what is a synapse? You guys know what a synapse is from the lecture about action potentials, but what is it? What does it connect? It's like written right there. Basically, a synapse connects one neuron to another, or one neuron to another cell. What the other cell could be? It could be a skeletal muscle cell, okay? So basically, the end of an axon, it's gonna have to connect to something. Where it connects, that space between them, that's a synapse. So whether it connects to another neuron by its dendrites, because we said the dendrites are the ones that receive information. So if you have an axon to a dendrite, this space between them is called a synapse. If you have an axon that connects to a cell, this is the neuromuscular junction, this is still a synapse, okay? So it's basically the site of the impulse transmission. What do I mean impulse transmission? Okay, so the entire physiology of like how action potentials work is that it goes from electric signals and it turns like when it's being released, it's released as chemical signals. Okay, so basically it converts an electric signal, okay, or nerve impulse to neurotransmitter or a chemical signal. Okay, this is how it goes by to the other cell. You can think about it as that this is like a free floating space. Okay, you can't have electricity or like electric signal pass through that. It's just gonna like disappear. So you need it to be chemical for a second. Okay, so when it's passing through here, these are the neurotransmitters. Okay, some examples of neurotransmitters that you guys know is like dopamine, serotonin. Yeah, these kinds of stuff. They're all neurotransmitters, and they are the transmission of signal from an axon to the efferent muscle or another neuron. Okay. Now you have three components of the synapse, okay, or the synapse, okay. There's a presynaptic axon terminal, or the terminal buton. Okay, I don't know how it's pronounced, but this is how I pronounce it, buton. But basically, presynaptic. So we said the synapse is the area between them, okay? So anything before that is presynaptic. Anything after that is postsynaptic, okay? So what identifies the presynaptic? Okay, it contains the mitochondria and it contains the synaptic vesicles where it stores the neuro transmitter. Okay, so you see these vesicles and mitochondria. These are all found in the terminal buta, which is the end of an axon. Let me see. Yeah, the terminal butons. Okay, terminal butons, all of them. The end of an axon. This is how it ends. Okay, so the presynaptic contains what? Contains vesicles and it contains mitochondria. What's inside the vesicles are the neurotransmitter, because once it touches the surface of it, or once it's going to propagate, it's going to go by exocytosis and it's just like going to pass through. Now, the postsynaptic. What identifies a postsynaptic? You see these kinds of stuff, what are they? Just like sodium and whatever, what are they called? What are they called? The channels, yes, channels, or the ion channels, and or receptors, okay? It has both, like either or. So this is a channel, okay? And this is basically what's the lining of the postsynaptic. It makes sense, okay? We have the axon, it's gonna go. The action potential is propagating through it as a electric signal. Once it reaches the end of it, or the terminal buta, it's going to be converted into a chemical signal where it's going to be released from its vesicles into the synapse, okay? Now, once a neurotransmitter is released, we said, what is it? It's a chemical, okay? So chemical, it has to bind to something or pass through something. So it's gonna bind by the receptors, or it's gonna pass through the channels. Okay, it's very easy. Synaptic cleft is the one that separates them. Okay, so again, very easy. The end, the presynaptic is it's the terminal butin of an axon. Okay, postsynaptic is whatever cell it is, whether it's a muscle or a dendrite. It has the channels, it has receptors. Okay, easy. We'll move on. Where is the chant? Yes, okay. Okay, so this was a slide in your presentation, but it's just a very easy concept. Basically, there are kinds of synapses, okay? So look at this big neuron, okay? The neuron is in the middle. Hold on. Yeah, this neuron is in the middle, and it has what? These are the dendrites, and these are the axon. Axon, okay? So the first kind there is axosomatic, from its name. It's axosomatic. It's an axon that connected to the soma directly, okay? So this is possible. Like we said the axon could either connect to another neuron. This is a way it can connect to other neurons. It's either by axosomatic, where it connects directly to the soma and releases everything in it to the soma itself or the cell body. Second type is axodendritic. Like I know it says here exoxonic, but just to make sense, we're gonna go by axodendritic because we're in this space. Dendrites, okay? Axons connected to dendrites, okay? This is one of the examples that we said. So when it connects to an axon to axon, either connect to the soma itself, which is axosomatic, or it connects to the axodendritic, okay? The last part it could connect to is axoaxonic. This one is a bit weird or a bit like ironic because we said like axons connect to other axons. It's really weird, okay? But it happens sometimes. In very special cases, you can have axons connecting to other axon. It's not as common, it's not the but it is there. Okay, so just know that it's like a kind of are they types of synapses of axon? Yes, these are the kinds of synapses for axons, okay? For cells, it's very easy. It's just one kind of connection to the muscle cell. It's a neuromuscular junction, okay? Axon connects to a muscle cell. Between them is a synapse or a neuromuscular junction, okay? It's easy. It's just like the confusion comes in the axon. So again, it connects in three ways to an axon. We said axon to axon. How is it going to connect? Either dendrites, okay, axodendritic, or axosomatic, so the axon to the soma, or axoaxonic, which is axon to axon. There's a lot of axons, okay? But this is, it's like it was in the slide, so I wanted to include it, but it's not that high yield, honestly. Question, okay. So when viewing the structures of a neuron, the cell body or the soma contains the highly developed rough endoplasmic reticulum. We said that, okay? So in the soma, there are rough endoplasmic reticulums and free ribosomes. These are the nestle substances. There's a specific region of the neuron cell body that's characterized by the absence of this. What is it? What area? C, axon hillock. C. Does anyone have any other answer? Does anyone disagree? Let's see. Yes, it is. So the axon hillock is the only area in soma that does not have rough endoplasmic reticulums, okay? Yeah, you guys are doing great. Okay, we're done with the neurons, okay? So we're done with the neurons, okay? Next part is the glial cells. Remember when we said the nervous system, the CNS or the PNS, whatever it is made of two kinds or two families of cells, made of glial and neuron cells. We finished the neurons, everything tamam. Let me check. Is everyone good with the neurons? Do you all understand it? Yes. Okay, perfect. If you have anything you don't understand, let me know. I'll go back. Okay, perfect. So the glial cells now, let's go to it. Glial cells are the supporting cell, which basically means because neurons are so important, you know that you guys know that neurons are the only kinds of cells that don't regenerate, okay? So once it dies, it's dead, that's it. So because they're very important, they need some supporting cells, you know? So these kinds of cells are called glial cells, and they're different kinds of glial cells, okay? So they're going to differentiate it into two classes. One, the glial cells of the CNS and the glial cells of the PNS, okay? Or the ones that are found in the central nervous system and the ones that are found in the peripheral nervous system. What are they, okay? The ones in the CNS, you have oligodendrocytes, okay? You have astrocytes, you have ependymal cells, and you have microglial cells, okay? We're going to go into detail for each of them, but just know that they are CNS, oligodendrocytes, astrocytes, ependymal cells, microglial cells. These four are found in the CNS, okay? Now, the ones that are found in the PNS are the satellite cells and the Schwann cells, okay? We're also going to go into detail about what they are, okay? I think there's a question that I've topic, but are all brain tumors resulting from glial cell division? No, that's a good question, but there are some tumors that can originate from neuron cells themselves, okay? I forgot the name, honestly, but a tumor can come from anywhere. So no, it does not just originate from glial cells, although it is the most common kinds of tumor, okay? Okay, thank you. Okay, so these are the cells that we have, the ones in the PNS and the ones in the PNS. So, did I say PNS twice? CNS and PNS, okay? So let us start. Astrocytes, where did we say they were? CNS or PNS? CNS or PNS? CNS, yes, they are CNS. You guys are focusing, great. So basically, astrocytes, they remind me of like Astro Boy. I don't know if you guys know that movie, but this is how I remember them. Astrocytes, Astro Boy. So what is a function of astrocytes, okay? So they form something called the blood-brain barrier. This is what this is, BBB. It's a blood-brain barrier, okay? It's one of the most important structures in your body because it prevents like the passing from like infections or any foreign material or any like autoantibodies or anything from passing into your brain. It's a very, very important structure, okay? What are meninges? Meninges is a lining of the brain. Okay, so the thing that lines the brain is called the meninges. It's the lining. Okay, there's meningeal fluid or the CNS fluid. It's between the meninges and the brain. You're gonna take this in neuro, inshallah. But that's what meninges is basically. Perfect. So the lining of the meninges and the blood-brain barrier. These are astrocytes, okay? So think of them as like the soldiers. Actually, all glial cells are soldiers, but these ones, they have feet, okay? What do I mean they have feet? Their feet are the ones that are like their weapons. So this is a blood vessel, okay? Think of this is a blood vessel that is going to the brain or inside the brain, okay? Inside the meninges or whatever it is. So this blood vessel makes the blood-brain barrier, okay? What does it have around it? These are the foot processes of the astrocytes. You see this astrocyte? This is a clear picture. It has like these foot, okay? They're wrapping around the blood vessel, okay? Why? Why does it do that? Does anyone know why? What's the function of astrocytes? Why are they wrapping around it? 1 second. Protection. Yes, exactly. Protection and preventing any microbes from passing. Like I said, blood-brain barrier, when you think about it, it's the barrier. It doesn't want any bad things coming in. So one of the things that make the blood-brain barrier is the astrocytes. Okay, so they have their foot processes that surround the blood vessels, and they just like they're an extra line of protection. Okay, so what is a blood-brain barrier composed of? We said astrocytes. Okay, what else? Tight junctions between capillary endothelium. Look at this picture. This is very clear. This is a red blood cell, and this is what? This is an endothelial cell. An endothelial cell is a cell that makes up a blood vessel. Okay, it has tight junctions between them. Why does it have tight junctions? So that diffusion doesn't happen as easily, okay? So we just want to monitor whatever comes in and what comes out. So all these are going to contribute to that. So tight junctions between the capillary endothelium, there's basement membrane, okay? And there's astrocyte feet, okay? It's just like memorize it how it is. It could come as an SQ, it could come as a multiple choice, whatever it is. If it asks you, what are the components of the blood-brain barrier? You're going to say what? Tight junctions between endothelial cells, astrocyte feet, and basement membrane, okay? It's easy. Just memorize what it is. I think there's a question. What's a pericyte? The outer layer of the blood vessel. We're gonna go more into details about that. I think where did you see pericyte? Hello. Okay. Could you repeat the last point? Yes. So the last point was in the right. Where is it? Oh, pericyte. Okay. Yeah, it's like this is just one of the cells for that line the blood vessels. Okay. It's not that like not potent, what the hell? It's not that important for this lecture, but pericytes is one of the cells that line blood vessels. Someone said repeat the last point. The last point we were talking about the blood-brain barrier. Okay, the blood-brain barrier, why is it there? To prevent anything that's not supposed to be in the brain from getting there. Okay, so it's the one that lines the brain, the meninges. Okay, it's composed of what? It has the blood vessel. Okay, so what makes a blood vessel strong enough or protective enough to make it part of the blood-brain barrier? Three things we said: tight junctions between the endothelial cells. Okay, two is basement membrane. Three is astrocyte feet. Okay, did you get that? It's just like the aspects of the blood. Look at this. This is a vessel. It has astrocyte feet. Okay, perfect. خلاص. Can you explain astrocyte feet? It's what it is. It's an astrocyte. Okay, let's compare it to an axon. Not the same function, but the same look. You know how an axon has an axon and there's a terminal buton? This is basically the same. Okay, so the astrocyte is a cell that has like, these projections. Okay, and each projection ends with a foot. Okay, it's just called an astrocyte feet. That's what it is. And what it does, it just like wraps around it. So if you see that this water bottle, okay, and my hand's an astrocyte, it does this. My fingers, like the tips of my fingers are the feet. So this is how like it attaches to it. You get it? Is it clear? Okay, so another aspect, okay, perfect. Another aspect for astrocyte is that they make astrocyte scar. This is not important for you guys at this level at all, but just like later on when you hear about it, some infections are, or like some diseases are caused because too much scarring, okay, in the brain. So one cell that makes a scar in the brain is astrocyte. It's good. It's good in terms of healing. Okay, so when you have a wound or anything, you want it to scar so it doesn't like stay open. But sometimes it can be dysfunctional. Okay, you don't have to know that. I'm just telling you just to like you can wrap it up in like clinical aspect. Astrocytes, they help in making scars. Okay, so if you have a question, what is a function of astrocytes? Give me one function of astrocytes. One. Healing. BBB. Yes, exactly. So blood-brain barrier and healing. That's it. We're done with astrocytes. All good? Protection. Yes, that's also protection. Great. Perfect. Okay. Now, the second cells are oligodendrocytes. What was it a cell of? CNS or PNS? CNS, exactly. Yes. Oligodendrocytes are cells of PNS. Now, this is very important for you to differentiate PNS cells and CNS cells, okay? Oligodendrocytes are only in the CNS. Why? Because these are the myelinating cells for the CNS neurons, okay? That's the most important part of it. So look at this diagram, okay? You have the CNS, the central nervous system, and you have the peripheral nervous system, okay? This is the neuron, this is a cell body, okay? See this cell? It's an oligodendrocyte, okay? Now the ones in the PNS are called Schwann cells. We're gonna go to them like in a bit, like just a bit, but we're gonna first start with the oligodendrocytes. These are the ones that cover or myelinate the axons of the CNS neurons, okay? So it makes white matter. What makes white matter white? It's a myelin. Myelin sheath, when there's a lot of it, it makes it look white. Okay, so whenever you see something as white matter, so unlike the spinal cord or in your brain, if it's white matter, it means it's axons. Why? Because it's myelin. And what are myelinated? Axons are myelinated, okay? Guys, you cannot myelinate dendrites. You only myelinate axons, okay? So if there is myelin or there's a white matter anywhere, it means there are axons and myelinated. There are myelinated axons. Got it? Okay, the other point is that one oligodendrocyte can myelinate up to 60 axons, okay? What that means, just think of okay, from the name, it's a big name. Oligodendrocytes, it's a big name. It means this cell is huge in terms of like its function or how many cells it can attach to. So one oligodendrocyte, it has so many projections, they can actually wrap around more than 60 axons, okay? This is very different from Schwann cells because Schwann cells, one Schwann cell can only myelinate one part of an axon, okay? So look at this. This myelin, it's a Schwann cell. The oligodendrocytes, this is one. I don't know if there's a better picture. There is not a better picture, but one oligodendrocyte can myelinate so many axons, okay? So just like, it's not in one oligodendrocyte to one axon, no. This is a Schwann cell. One Schwann cell, one axon, okay? Now, again, myelin sheath is a modified glial plasma membrane wrapped around the nerve axon, okay? It's just a fancier way of everything that I said. It's basically you have an axon, it's covered by the glial plasma membrane. What is a glial cell? Could be either an oligodendrocyte or a Schwann cell, depending on where you are, okay? Schwann cell is found in the periphery, oligodendrocyte is found in the central, okay? Now, the road of Ranvier. Someone asked a question about that before, and you were comparing it to the muscles, okay? But now this is the actual road of Ranvier, okay? It's the gap between the myelin sheaths that does not have myelin. So you see this area right here, this one, this one, this one. These are called the nodes of Ranvier, okay? Now, the entire pathophysiology of it is like, it's too much details, but what you basically need to know is that it allows saltatory conduction. I think Abdul Jabbar said it before, because whenever I say that word, I just remember AGR's voice in my head, saltatory conduction. It basically means that the action potential jumps, okay? It's like it hops on. So it's a faster action potential. That's basically what it is. It's like instead of the action potential taking the entire axon, and guys, neurons, by the way, or neuronal axons are so long. It's probably one of the longest cells in your body, because one nerve, like for example, if you think about the what was it called? If there's a nerve that runs from your spinal cord to the top of your toe, okay, this is an entire nerve. It doesn't like break down. No, it's just one full nerve. So an axon is so huge, okay? So imagine the action potential passing through all of that. It's gonna be like, nope, I'm not doing that. So what does it do? It jumps. It jumps from the roads of Namvier, from one node to another node to another node. So instead of taking the long way, it just jumps. It just goes, it skips a couple of meters, or it skips a couple of steps, okay? This is why it moves faster, okay? So it's called saltatory conduction, which basically means the action potential jumps from node to node, which causes a faster conduction of the action potential, okay? Easy. Dun, dun, dun. Okay. Yes, perfect. Okay, this is another picture of it. Oligodendrocyte, look at it. One oligodendrocyte is like, it's holding on to two. Schwann cell, each and own Schwann cell by itself. كل واحد وحده. Okay, it's like alone. It's just one cell, one part of the axon. Oligodendrocytes, no, it could hold more than one. Like we said, up to like 60. Now, this is like how a myelin actually looks like. It's like if you cut it across section, okay, and you look at it, it's just like onion skin. You know, it doesn't look that much in microscope, but as a like a concept, it's like onion skin. It's just multiple wrapping around each other, okay? It's like if you take a piece of paper and you roll it, okay, and you look at it from the center, this is what it is. It's like a myelin, okay? So this is just everything we discussed. Okay, now this is it for the oligodendrocytes, okay? Next are the microglia and ependymal cells. These are the last cells that we have from the CNS cells, okay? So microglial and ependymal cells are found in the CNS, okay? What are they? Now, these are like very easy, like I only put them in one slide because they're honestly very simple. Basically, microglia, they originate from macrophages. What does that mean? If a cell originated from macrophage, what is the function of a macrophage, or what does it do? Yeah, who knows what a macrophage is and why microglia, phagocytosis, exactly, that's the word I'm looking for. Immunity, phagocytosis, all of that. This is what a macrophage does. So imagine you have a cell that's derived from a macrophage. What does it do? It does macrophaging too. It does phagocytosis, yani. But where in the CNS? Remember, okay, here we're gonna tie some concepts together. Remember, we said the blood-brain barrier only allows certain things to pass through. This also includes cells, okay? So macrophages are not welcome in the blood-brain barrier. They're like, no, I don't like you. We're gonna have our own macrophages, and we're gonna call them microglia, okay? Microglia comes from monocytes. Comes from monocytes. If I'm not mistaken, yeah, I forgot, honestly. This was in the lecture from him, the whole cycle of it. I honestly forgot where it derived from, but you should check it. You guys know better than me in that aspect because I haven't studied this in a while. But just know for microglia, they are derived from macrophages, okay? So macrophages, they do phagocytosis. So what do microglia do? They do phagocytosis too, in the CNS, okay? Very simple. Now, the ependymal cells, these are very important cells because we took them again in neuro in second year, okay? Ependymal cells are the cells that release or they assist in the production of CSF. What is CSF? Cerebrospinal fluid, okay? The cerebrospinal fluid, which is the fluid between the meninges and the brain, or like the fluid in the CNS everywhere, this is what makes it, the ependymal cells, okay? You're gonna take the like how it does it exactly and its whole like what's it called? like the whole process of it later on, inshallah, but just know for now, like at your level, you just need to know ependymal cells are the cells that release cerebrospinal fluid, okay? And they're found where? They found around the ventricles or the central canal. Now, ventricles, don't be confused from ventricles of the heart. I'm not talking about ventricles of the heart, I'm talking about the ventricles of the brain, okay? In your brain, inside it, there are ventricles, okay? Why are they there? Because it's where cerebrospinal fluid is secreted and stored, okay? So the cells lining the ventricles are the ependymal cells, and they're the ones that make the cerebrospinal fluid. So look here, this is a capillary or a blood vessel, and these are the linings of it, okay? It's a columnar or cuboidal cell with no basement membrane, okay? It's from the histology right here. You see them, they're all columnar. Some of them are cuboidal, but they're columnar slash cuboidal cells with no basement membrane that line the central canal or the ventricles of the brain. Central canal is the one that passes right in the middle of the spinal cord. Okay, it also contains the CSF, and this is where we do the lumbar puncture. Okay, so again, whenever you see microglia, think of what you think of macrophages. When you see ependymal cells, you think of CSF. Okay, easy. This is the last part for these CNS cells. You guys got it? Yes. Perfect. Okay. Now the glial cells of the PNS, there are only two. Okay. One of them is a Schwann cell, the other one is a satellite cell. Okay. Now, Schwann cell, what was its brother or the sister or whatever, the other one of it was an oligodendrocyte. We said that oligodendrocyte is found in the CNS. Okay. Now the Schwann cell, this is the Schwann cell. The myelin that it makes is itself. Okay. So imagine the cell, like, you know, Elastigirl from The Incredibles. Imagine the cell is Elastigirl and just like expands so much, it makes itself into a thin sheet and it wraps around the axon. This is basically a Schwann cell, okay? It's just one cell that myelinates one part of an axon, okay? It's found in a PNS, one Schwann cell, one portion of an axon. And it also has another function, okay? So don't think that it only myelinates cells. No, it has another function, which is it just basically supports non-myelin neurons. So this is not very high yield, but just in case like it came to you as a concept or if any of you read about it, Schwann cells support non-myelinated neurons and just like it just holds them together. This is not myelinated. So these axons are not myelinated. They're just supported by the Schwann cells. Okay, so don't be confused. Okay, don't get confused now. Schwann cells, when it myelinates, it's myelinating the axon itself. Okay, it wraps around the layer, it makes myelin, and this is how it looks like. Okay, it's a cross section. In the middle, this is the what's it called? This is a neuron, okay, and it's myelinated. This is it, okay? So the Schwann cells, it myelinates the neurons, but also has another function that it supports non-myelinated neurons, okay? It does not mean myelinates them, but the one that myelinates, it's myelinating, okay? So again, it's very easy. One Schwann cell, one aspect of an axon, okay? Now satellite cells are, okay, they form a thin layer around each large neuronal cell body in the ganglia. This is the keyword, in the ganglia. So if a question comes to you and it's like, what kinds of cells are present in the ganglia of the PNS and they provide structural and metabolic support, these are the satellite cells, okay? By the way, I'm sorry, I don't think these are myelinated. I can't find any myelin. I think these are just neurons. Yeah, these are just neurons, okay? So where is it? Here are the satellite cells, okay? You remember when the axon, like the axon itself, it has its nucleus in the center, or like the neuron itself. On the periphery, what does this remind you of? It reminds you of astrocytes, right? So this is very similar to astrocytes, but it's found in the PNS, okay? So you have an axon and the ganglia. What is a ganglia, by the way? You guys, I don't know if you guys know what ganglia is. Could you repeat the last point? I'm actually confused. Okay, no worries, no worries. Okay, let's start again. Basically, Schwann cells, okay? They are the other relative of oligodendrocytes. You guys understood oligodendrocytes, right? That it like myelinates axons and everything. If you understood that, take that and give it to the PNS, okay? CNS is very, very, very important and it's like a lot and has so many aspects. That's why we need a cell that is huge and it has so many processes that it can myelinate a lot, okay? The PNS is just like it's chilling, okay? It's living its life. It doesn't need that much support. So the cells that myelinate it are called Schwann cells, okay? Schwann cells, they wrap around one aspect of an axon, okay? So like for example, this is a pen. This pen is an axon, okay? You see this cap? This cap, okay? Imagine like it has a hole here. This is a Schwann cell. The one cap is a Schwann cell, okay? This is how it does it. Now, if we were talking about an oligodendrocyte, it'll be like my hand, okay? And it's gonna wrap around, like one finger is gonna wrap around here, and then one finger is gonna wrap around here. You see? One cell, one oligodendrocyte, it like wraps around multiple axons, okay? But the Schwann cell is just the one cell, and it covers one part of an axon, okay? It's smaller, it's just one, it can't hold on to more than that, okay? Now, the idea of the non-myelinating Schwann cell, like I said, it's just an idea. It's not high yield at all. So if you guys, if someone didn't understand it still, Adi, don't worry. I just mentioned it for the sake of like, to be concise, okay? Non-myelinating Schwann cell, basically it's a kind of Schwann cell, or it is a Schwann cell that has another function of supporting a neuronal axon, okay? So it's not called myelinating. This is not a myelinated axon, okay? This is just a supported axon, okay? It's not, you're not gonna get any histology picture of this because I didn't find any in the slides, صراحة. It was mentioned in just like a split second. So I just mentioned it in, it has another function that it supports non-myelinated axons as well. So in a PNS, one axon requires many Schwann cells. Yes, exactly. In a PNS, one axon requires many Schwann cells. Like the picture here. See, this is a Schwann cell, all of them. Okay. Non-supporting aspect. Is that clear? That like it has another function that it supports non-myelinated. It's not myelinating, it just supports it. Okay. Okay, perfect. خلاص, alhamdulillah. Okay, so this is it for the glial cells with PNS. We said the Schwann cells and the satellite cells. Someone asked, wait, about the ganglia, right? Ganglia, you guys took, I think you took, remind me if you took it or not. Basically, there was an aspect of neurology, like the what's it called? I think I have a picture of it here. Yeah, like this picture. Did you guys take it before, like the ventral horns and dorsal horns and these kinds of stuff? Yes, okay. So, do you know the dorsal root ganglia? The dorsal root ganglion? This is what I'm talking about. So inside it, it has satellite cells. You get it? This is what I mean, and they're found in ganglia. They're found in the dorsal root ganglia or whatever it is, you know? So these are basically the Schwann cells. Perfect, okay, alhamdulillah. Yeah, so they're found in the PNS. They're usually found in the ganglia. They provide support and metabolic support. That's it. They're supporting cells. So we have ganglia only in the dorsal root. No, not only in dorsal root. You have ganglia otherwhere. There's other examples of ganglia, but the one I'm talking about like now is the dorsal ganglia. Ganglia is just like one, it means a collection of cell bodies or a collection of like neurons clumped together, okay? Ganglia is in ganglia is in PNS only. Okay. Is this part clear? Should we move on? Yes, okay, perfect. Okay, this is a histo of dorsal root ganglia. So if you get this one and it asks you what kinds of cells can you find here, you're gonna say what? You're gonna say satellite cells. Exactly. Okay, perfect. This is how they look like. Again, just another picture. They're right here, right here. Okay. This is another schematic of Schwann cells and how they wrap around it. Okay, like I said, Elastigirl is just like expanding to a sheet and it hugs around the axon multiple times until it's completely isolated. Okay. Next. Okay, this is under the microscope. Again, I'm showing you guys a lot of pictures because in the OSP, it's going to come as pictures. So you need to be familiar with them. So this is a longitudinal section. This is a cross section. Okay. It's very easy. You get cross section. You cut this one in half. You look at it from here. It's gonna look like I don't want to spill water, but it's gonna look like round hole. If you cut it this way, it's gonna look longitudinal. Okay, so longitudinal cut and the cross sectional cut. Yeah, right here too. Okay, now the last part for this is a neuropil. Neuropil is basically how can I explain this? Neuropil is basically the everything else. So you can't have neurons floating around. So you can't have them floating. Where are they? They are suspended in the neuropil. So it's a network of fine cellular processes emerging from the neuron and ganglia. What does that mean? So what does a sentence mean? It's a network of fine cellular processes. It's everything. Okay. صح, neurons, they have dendrites, they have axons, and then حتى astrocytes, they have feet, and everything has extensions, okay? These ones, when you put them all in one bowl or like one place, it's gonna fill an area, okay? So everywhere is gonna be like a lot of lines. Think about it as a spider web, just a very thick and heavy spider web, okay? So that all the extensions of all the cells found in the CNS or in the PNS, they're all suspended in the neuropil, okay? It's associated with the gray matter, okay? Like we said, white matter was myelinated, so gray matter was a non-myelinated, and gray matter contains what? What does gray matter contain? So axons, yes, axons and dendrites of the neuron, and also like from other cells, any processes. Guys, what does gray matter contain? We said white matter contains soma. Yes, it contains cell bodies. So like I said, because the somas have like dendrites and everything like surrounding them, the neuropil is usually found in the gray matter. Okay, so you see gray matter, it usually has the neuropil. Okay, doesn't mean it's not anywhere else, but it like where it is mainly is in the gray matter. It's not visible in H&E stains, so you're not gonna really see the neuropil here. It's just gonna look like a bunch of like shakhabit, okay? Here you can see them more. So see how the dendrite is like has so many processes. This is where it is basically. So neuropil, think about it as a space, the space that supports it. So the axons and the gray matter, yes, the axons and the gray matter are unmyelinated, where they're myelinated in the white. Okay. Now this is a part I think you already took it, but it's like just let's go over it because it was a slide. White matter and gray matter. Okay, gray matter contains what? Contains neuronal cell bodies. The soma, like you said, has dendrites, has astrocytes, microglia. It's basically where the cell bodies are and everything surrounding them. White matter contains myelinated axons. It contains oligodendrocytes if it was in the CNS. If it was in if it wasn't, it was Schwann cells. It contains astrocytes and microglia too. Okay, and very few neuronal cell bodies. Okay, I think there's a question. So we have axons in agreement. Yes, we have axons in agreement. We have axons everywhere. Okay, because the nervous system is made of neurons. So you have axons everywhere. But the axons that are in the white matter, they're myelinated. If the axons are in the gray matter, they're non-myelinated. Okay? Does that make sense? Clear. Perfect. Okay. Is this part clear for you guys too? You know, white matter consists of basically oligodendrocytes or Schwann cells if it was in the PNS, but this is for the central. Okay. Now the spinal cord, same thing that we just mentioned, okay? You guys took this before in anatomy, so I'm not going to go into detail. But basically, you have the dorsal horn, the ventral horn. The ventral horn is the one that contains the motor neuron. The dorsal horn is the one that contains the sensory neurons, okay? Near the center, you have the central canal, okay? Now, question. What is found in the central canal, okay? What is found and what makes it? This is a very, very, very OSPE question. Yes, it contains CSF. What makes a CSF? Ependymal cells. Perfect. Yes, exactly. I think I'm pretty sure this came in the OSP at some point, but it's gonna like point an arrow into the central canal. It's gonna be like, what lines this? You're gonna say ependymal cells. What does it release? It releases CSF. That's it. Clear and simple. Okay. Yeah, this is just another picture, and you have Schwann cells. This is the ganglia. This is mainly anatomy, so just like, yeah, that's it. Peripheral nerve, like I said, it's similar to a muscle, you know? So you have like the epineurium, it has, but just like know if a peripheral nerve, then it is myelinated by a Schwann cell. If it is myelinated, it's going to be myelinated by a Schwann cell. Okay? These are the coverings of them. So remember in the muscle, there was the epimysium and the perimysium and the endomysium. Same thing for a peripheral nerve. Okay? It has endoneurium, it has perineurium, and it has the epineurium. Okay? Same thing as a muscle. Okay, epi1 is the dense outer connective tissue that connects the entire nerve. Okay, so you have this entire nerve. So for example, this is like a nerve. It's surrounded by the epi here, epineurium. So this whole nerve, epineurium. Okay, if you go inside the perineurium, it's the one that surrounds group of axons and Schwann cells, or the endoneurium bundled together. Yeah, basically, if you have an axon with its Schwann cell, they're each going to be covered with a perineurium. Okay, you get it? And also, just so there's no confusion, when I say it covers an entire nerve, the nerve itself has multiple like nerve cells. Okay, so a big nerve has nerve cells. So that's why I said if it covers the big nerves in the one that you actually touch and see, like when you see an anatomy, now this nerve, it's covered by epineurium. So if you have a question in the OSPE and it points to a covering, a dense connective tissue covering of a nerve, the big one, it's called the epineurium. Okay. Perineurium is the one that's around the axon and its Schwann cell, if it was in the PNS, but we're talking about the peripheral nerve. Endoneurium is the one that's connecting each axon plus its one, like basically like each one by themselves. Okay. So that's how when it comes to endoneurium and perineurium, it's not that high yield because lines blur when it becomes an endo and when it becomes a peri. So just know that the epineurium is the one that covers the entire nerve, the big nerve. Okay. Now, Do you guys know something called vasoviscorum? Do you guys take that? Vasoviscorum. Does anyone know what that is? Okay, basically it's the arteries that supply the arteries of the heart, okay? If I'm not mistaken, inshallah. So we have something called the vasa nervorum, or it's basically the cells that supply the peripheral nerves, okay? Because again, nerves are still an organ. They need their own blood supply. So what supplies them is called the vasa nervorum. Wait, there's a question. White axon makes CSF2. No, not white axon. What makes CSF is ependymal cells, okay? CSF is made by ependymal cells, okay? Epineurium, the entire thing, and peri covers individual fibers of that nerve because this always confuses me. Yeah, it confuses me too. But yeah, it's exactly what you said. Epineurium covers the entire nerve. This entire nerve is covered by epineurium. Look at this drawing. Okay, let's see if it's clearer here. Endoneurium, see, like this is a group of axons. Like this one is on between them. Perineurium is like everything with them. It's very confusing, Salaha, because like I said, the lines blur between them. This, okay, here's an axon, Schwann cell, okay, all of this. Around them is a endoneurium in between each of them. Around them as a whole, perineurium. Okay? Around the entire nerve is an endoneurium. I think it's still confusing, right? Honestly, guys, for this entire drawing or like the peripheral nerve and the coverings, just know the epineurium. This is the most important part. What is a vasa nervorum? Vasa nervorum, like I said, they are vessels that supply the what's it called? The nerve itself. Okay, that's just what they are. They are small vessels that supply peripheral nerves. Okay. Um, can you go back to ependymal? Ependymal. Okay, yeah, ependymal cells. Sure. Where is ependymal cells? Did I pass them? Yeah, ependymal cells. These are epithelium-like cells. They're either columnar or cuboidal that line the ventricles of the brain or the central cord. Okay, what do? They release CSF. So, ependymal cells know that it releases CSF and it also lines the ventricles of the brain and also lines the central canal. Okay, is that clear? It's BFN, BFN, BVN. I'm sorry, I don't get that. Can you explain what's BVN? Where were we? I mean, vasa nervorum. Vasa nervorum is the vessel that supplies the nerves. Okay, the peripheral nerves. What supplies them is vasa nervorum. That's it. Don't confuse it anything. If you have OSP of a nerve, okay, and it points at it and it says what covers it, what covers it? What's covering the peripheral nerve? What's covering the peripheral nerve? The big covering, the entire nerve. What's it covered by? Endoneurium. Okay. Now, if I get you a second part of a question and I say, what kind of vessel supplies it? Blood vessel. What supplies vasa nervorum? Yes, exactly. You get it? This is how it might come. It's a very straightforward question that they can actually get it in. It'll be like, what supplies it? Vasa nervorum. Clear? Okay, let's have a question. Okay, which glial cell type in the central nervous system is derived from circulating blood monocytes and functions primarily to protect the CNS by engulfing infectious agents? D, microglia. Yes, amazing. Does anyone disagree? Let's see. Yes, it's a microglia. Like we said, it's derived from macrophages. Okay, and according to this, whoever said that it's derived from the monocytes, it's correct. It's derived from monocytes. Okay, microglia. Yes, microglia, they are from the circulating blood monocytes, which are the macrophages. They're the ones that protect it, phagocytosis, all of that. This is the microglia. Okay. This is it for the nerve. Okay, now we're at the last part of this pal. Okay. Microglia is only in BBB. No, microglia is in the CNS. It's everywhere in the CNS. Okay, remember we said there are CNS cells and there's PNS cells. Microglia are found everywhere there. It's the phagocytic cells. Okay, they are the soldiers. They're the ones that fight infections. Okay. Okay, good question actually. What cell was the one that made the blood-brain barrier? What was it called? Astrocytes, exactly. So don't confuse it, okay? What's it called? Microglia does not make the blood-brain barrier. It's everywhere in the CNS. It's the macrophages there. It's the one that like cleans up the debris. Astrocytes are the one that make the blood-brain barrier. Okay? Is that clear? Okay, is everything clear for the nerves lecture? I know there was a couple of hard topics here, so if you guys have any question, let me know now before we move on to the lymph, which is the last part. We finish this and y'all are free to go do whatever. Okay, yes. Okay, let's move on. Okay, so lymph. Microglia is in the brain and then moves from there. It, it moves everywhere in the CNS. So it moves like from the brain to this, it doesn't move. It's just stuck where it is, but it's found everywhere in the CNS. That's a better way to put it, okay? So microglia is not found in your normal like blood vessels. It's not found there. It's only in the CNS. Okay? Like I said, remember when I told you the CNS are very important and they need their own bodyguards. So in our body, we have the macrophages. These are like the protective cells, the immune cells. In the brain, they don't like the macrophages. They have beef with them. They don't like them. So they have their own macrophages, which are called microglia. Okay, so in the CNS, we have microglia. Does that make it clear? Perfect. Okay. Now for the lymphatics. Okay, lymphatics, صراحة, I'll tell you guys one thing before we start. There was one question in the OSPE from the lymphatics, which was, it was the histology of a thymus. Okay, we're going to talk about the thymus gland right now, but it was a histology slide of a thymus, okay? And I studied this lecture very well, okay, because صراحة was very interesting to me. So I knew everything. I knew the inside out of the thymus, okay? Like the histology of it. I knew how the cells looked like, everything about it. But when it came to the question, the first question from the in the OSPE, what is this organ? The second I saw the picture, I knew what it contained, I knew what's the function, everything, but I forgot that it was called a thymus. So please don't make that mistake. Okay, when you're studying, study the picture as a whole. Okay, you need to know that this is a thymus and this is how it looks like from the inside. Okay, because that was honestly a mistake that I still remember now in third year, that I lost a point because I forgot the name of the organ. I knew what it was, I knew what it did, and I knew the cells, I knew everything about it. Well, I knew everything about it. But when it came to the name, I forgot the name. So I lost the entire question because of that. So please, when you're studying, study the whole picture. Okay, I showed you like pictures of muscle cells and everything. Why was I saying skeletal muscle cells? Because I don't want you to just look at the picture and be like striations, integrated discs. No, I want you to think this is a cardiac muscle, so it contains this, this, and that. You get me? Okay. Now let's actually start with the lymphs. Okay, so the lymphatic system is the immune system. When we're talking about immune system, that's it. That's what it is. This lymphatic systems. Okay, it contains lymph or lymph, like the vessels and everything, and the organs. Okay, lymph is a fluid. So if you look at this, so we have an entire vascular system. We have arteries, we have veins. Now with them, you have the lymph, the lymphatic system. Okay, would it be a good idea to use these PAL recordings in these slides to study? Yes, it would. I use them. I've been using them since first year, honestly. And PALs are made for you guys, so it's like a final revision, okay? So for example, for me, for my slides, I included all the important stuff from your lecture, okay? Everything that the doctor mentioned is important. The main concepts, the pictures, I used all the pictures from the slides. So I'm not gonna say it's enough for like the entire studying of it. So don't like, if you don't have time, ماشي, it's fine. But like it's mainly use it after you're done studying and you want خلاص, like you're done and you're gonna start solving questions, or you're gonna خلاص, like revise, use them as a revision. Okay, this is what I do till now in third year. I study the lecture, I listen to the doctor, I attend the lecture, and then when I actually want to study or there's stuff I don't understand, I go watch the PALs. Okay, are all PAL slides accessible? Yes, they're all accessible. I'm pretty sure you guys are sent a link either an email or something. If not, I'll ask Lema inshallah if like where do they send it to you. Okay, back to where we were. Lymphatic system, lymph, and organs. Okay, now what's the function of a lymphatic system? We said it's immunity. Okay, what is immunity? Immunity means fighting pathogens. Okay, so it responds to pathogens. Okay, whether it's by phagocytosis or whatever it is, and it kills viral cells. This is another important aspect of lymphatic system. Sometimes cells that are infected with viruses, they don't die. They just say viral cells, okay? So your body sometimes has no way of identifying them except by the immune system. It's gonna kill these cells. It's gonna like go to them and like destroy them. Another function of the lymph system is transport proteins from the diet. Okay, so you're gonna learn that I think in what's it called? Biochem. Okay, one of the pathways for like cholesterol metabolism or all of that is that the fat or like the fatty acids or everything that you eat from food, it gets first absorbed through the lymphatic system. Okay, so one function of it is transport lipids from the diet. Okay. And it also collects interstitial fluids into the capillaries and vessels. Okay, this is important for what kind of disease is edema. Okay, do you guys know what edema is? Does anyone know what edema is? Or have you ever heard of edema? Yes, swelling, accumulation of fluid. Yes, exactly. So when you have edema, there's a lot of diseases regarding like edema. It could be like a side effect of like heart failure. It could be something genetic. But basically, the whole physiology of edema is a dysfunction in the lymphatic system. Okay, because it contains the interstitial fluid. If there's anything wrong with the lymphatic system, it's gonna leak. When it leaks out, it's gonna be there between your cells. Okay, and it's gonna cause swelling of whatever organ it leaked to. Sometimes it can affect the entire body. Sometimes it affects just the lower limbs, like in heart failure. بس يعني okay, so Lema said that there is a group chat description. The past slides are linked there. Okay, so basically these are the functions of the lymphatic system. The transport lipids can collect interstitial fluid and they respond to pathogens and kill viral cells. Okay, so we have some classifications. Okay, the lymphatic system itself, it's classified into two. There's tissue and there's organs. Okay, the tissue, there's two kinds of tissue. There's diffuse tissue and there's nodular tissue. Okay, we're gonna go into details. What does that mean? But the other classification of it is organs. Okay, what do I mean by organs? There are two kinds of organs. There's primary organs or primary lymphatic organs, and there's secondary. Okay, primary is the bone marrow and the thymus. Okay, why is it called primary? Is because this is where the lymphocytes are made. Okay, so like T cells are made in the thymus, and the B cells are made in the bone marrow. Okay, this is where they originate. Okay, so that's why they're called primary. It's the first organ, and it's found since like embryological life. So when you're born with them. Secondary lymphatic organs are like the lymph nodes, the spleen, the tonsils, and any diffuse lymphatic tissue. Okay, so you know your tonsils, you know the spleen. All of these are the secondary organs. Okay, so the only primary organs are the bone marrow and the thymus. Why? Again, because these are the location for where B cells and T cells are made. Okay. Okay. Now, the supporting network. What does that mean? It means that everything has a supporting network. So we said in pop question. We said the supporting network for the nervous system. What was it called? Remember the neuropil, exactly. Neuropil was a supporting system for the nervous system. So the one for the lymph, it's basically made from reticular fibers, okay? You guys probably took it with Firas for the PAL about like the connective tissue. The one kind of connective tissue is reticular cell. May you go back for a second, please? Sure. Here. Is there something you didn't understand? This is just where we classified them, everything right. Okay, perfect. Okay, back to supporting network. So we said it's like the nervous system, but this one is made of reticular cells and reticular fibers. Why did I not say reticular cells and fibers? Because some organs, we're going to discuss now, contains only reticular cells, not fibers, okay? So you guys need to differentiate that. When I say reticular cells, I mean reticular cells. I don't mean reticular fibers, okay? It's very different. Reticular fibers are type 3 collagen, okay? And yeah, so when they're supporting it, it can either be reticular cells or it could be reticular fibers, depending on the organs, okay? The thymus only has, yes, exactly. Thymus has only reticular cells. You spoiled it. It's okay. Cells of the immune system reside inside this network, okay? What a network is for, it's a mesh network. Mesh means, reticular means mesh, okay? So what are reticular cells? So if you get a question about that, these are the reticular cells. Right here, you see them? This is a reticular cell, this is a reticular cell. It's elongated cell. It doesn't, remember how we said smooth muscle was fusiform? This is not fusiform, it's just elongated. So this is like a nucleus, and this side is cytoplasm, and this is cytoplasm. It's like a very long, okay? It's palely stained nuclei. It's not, see, it's not that eosinophilic. And lightly basophilic cytoplasm, okay, basically it makes the type 3 collagen, which is a reticular fibers. These are reticular cells, okay? Just be able to identify them. So if you see a picture like this in the OSP, صراحة guys, it's not يعني that clear, like why is it reticular cell, but this is one of the things that you just need to be able to identify. So you see something like this, okay, it's usually going to be zoomed in. First identify where are you, so which organ is it? So if it was like thymus or a lymphatic organ, you know it's going to be a reticular cell, okay? And if it says what are like the identifying features, say it's an elongated cell with palely stained nucleus, and it also synthesizes type 3 collagen or the reticular fiber, okay? I think there was a question. Do reticular cells and fibers make the supporting network or the opposite? Yes. It's entire system. Reticular cells make reticular fiber, and them together or not together are their supporting network. Okay. So the supporting network could be either just reticular cells with no fibers, or it could be reticular fibers with some cells. Because you need to have the cells there. If you have the fibers, you need to have the cells, because what made the fibers, you know? So the supporting network is made from reticular cells or reticular fibers with the cells. Clear? Just know these pictures. This is the supporting network. Okay, now the lymphatic vessels. Remember when we said you have the vessels like the arteries, you have the veins, you also have the lymphatic vessels, which is very important, okay? So the identifying features for these are what? They have valves, okay? What also has valves? Do you guys know what kind of blood vessel also has valves? Is it an artery or veins? Yes, exactly. Does anyone know why? Why do veins have valves and arteries don't? To stop backflow, exactly. Because the pressure inside veins is not as high as an artery, so usually blood tends to backflow, which causes like a venous insufficiency or like other diseases. So it has valves. Same thing with the lymphatic vessels. It doesn't have actually any like pressure or like very, very, very low pressure because it doesn't have a heart. Veins are pumped, okay? Like, so the blood inside the veins is being pumped by the blood. It just doesn't have that much pressure. Lymphatic vessels don't have any pressure. It doesn't have a pump, okay? So it needs valves. So there's valves in the capillaries. There's valves everywhere in the lymphatic vessels, okay? Yes, so they're similar to the capillaries, but what differentiates a lymphatic vessel from a capillary is that there are no tight junctions, okay? So you see here, it's just like it's pores, like the cells are on top of each other. Like this cell, this cell, this cell, this cell, they're all on top of each other. Capillaries, usually they're like right beside each other, and where they attach, there's like some tight junctions or anything that connects them, okay? So why is there no tight junctions between the cells of the lymphatics? Why is why is it just like there? This is very floppy, okay? Why do we need it like that? General question, do we need to know the diameter to allow diffusion? Exactly, yes, to allow diffusion. 1 second. Do we need to know the diameter, dimension of the cells and all? No. I know it's like in the slide, sometimes it says like 40 to 50 millimeters or micrometers or whatever it is. Honestly, we've never had a single question about them. If you want, for the sake of like cohesiveness or whatever, yani, you can memorize them if you have time. If you don't, صراحة, we never had a question about it before. Allow diffusion of interstitial fluid. Yes, exactly. Sahwi said no. Edema is caused by dysfunction. Okay, so yes, we have pores and we have spaces between them, but when it comes too much, this is what causes an edema. So normally you have pores between them. You have no tight junctions between the lymphatic systems, okay, which is what allows interstitial fluid to pass through, okay? It's not normal if it's too much, okay? It also has fine filaments or like that anchor them to this connective tissue, okay? It's just like what it is, like what anchors it to the connective tissue, fine filaments, okay? This is basically saying that it's not floating freely, okay? It's anchored to the connective tissue right underneath it by lymphatic vessels, okay? So from this picture right here, this is an important picture. I remember it was in some sort of quiz, I think it was a TBL. This is an arterial, this is a venule, and this is a lymphatic vessel, okay? شوفوا the difference between them. Arterial has a very thick wall. Veins don't have a thick wall and they're usually like irregular in shape. This is the connective tissue. This is a lymphatic tissue. You see how it doesn't have like a very structured like surrounding of it? It's similar to a vein, similar to a capillary, but it doesn't have any, like we said, tight junctions. And like you see how it's like anchored to the connective tissue? It's anchored by the fine filaments, okay? So lymphatics, it doesn't have any blood vessels inside, by the way, guys. If you see blood inside any vessel, it's either an arterial or a venule. It's not going to be lymphatics unless there's a pathology behind it, okay? It's not normal to have blood in your lymph. Okay, 1 second. This is my cat, guys. Say hi. Okay, basically, like we said, there's no blood in lymph. No blood in lymph. So one thing that you can differentiate them like histologically is that if you see a cell or if you see any vessel or whatever it is, yani, that doesn't have blood and looks like that, it's like irregular, that is a lymph vessel. I think there was a question. It looks empty. Yes. Why does it look empty? Because it has no blood vessels. Okay. Now, one important fact about lymphatic vessels is that the only means that protein can leave the vascular compartment and return back to it is by lymphatic. This is what edema is by the way. This is what edema is. Proteins make the fluid more I forgot the name, but basically, we have more proteins in a vessel, it's going to cause water and fluid to pass to it, okay? It's going to go to it, okay? So if you have a dysfunction in the lymph, like we said, lymphs are the only ones that can move the protein. So if there's too much protein outside the lymphs, water is going to go outside it. So there's going to be edema. There's too much proteins inside, there's going to, the opposite of edema or whatever it is. Hold on. Yeah. Yeah, the colloid pressure, that thing, okay? Do you guys understand this? The blood vessel, the lymphatic vessels. As a summary, they have valves. They're the only ones that transport proteins, okay? And the difference between it and a capillary is that it doesn't have any tight junctions, okay? And the function of it is to transfer interstitial fluid. Okay. Now, this is a lymphatic circulation. Again, it's what we said. Like we have our normal circulation, the blood circulation, okay? I didn't get the protein thing. Okay, the protein thing is in another lecture, but I'll mention it again here. Basically, you have a lymphatic vessel, okay? The only kind of vessel that can transport proteins is lymphs, okay? Proteins, they shouldn't be in the blood. Or like when they do go to the blood, they just stay in the blood and you get excreted, okay? They don't go back into, like you don't transport proteins. It's either in the cells or they're outside the cells. When they're outside the cells, they're in the lymph, okay? So that's what I mean when it says it's the only way a protein can leave the vascular compartment and come back to the vascular compartment, okay? Now, if there's a dysfunction in that, if there's too much proteins that is outside the lymph, where protein goes or where salt goes, water follows. If there's too much protein outside the lymph, water is going to go with it, which is going to cause edema. It's going to cause a swelling because the interstitial fluid is not contained inside the lymph, okay? This is how basically the protein thing works. If you have too much protein outside the lymphs, if there's a dysfunction in the lymph vessels and the protein is not staying inside, you're going to have edema. Did you get it? Perfect. Okay. Circulation, like I said, this is our normal circulation from the heart to the heart, everything good. Now, beside it, what attaches to the side, you have the lymphatic circulation. Okay, this is what we said, like the water, the lymph nodes. This is the immune cell. This is how we filter our blood. Okay. Now, the nodules. Okay. Now, this is where it comes important. Okay, lymph nodules they're not constant, which basically means what? Sometimes, for example, in primary nodules, they're there when we're born. Okay, so the thymus, our thymus, okay, it's a full organ and everything when we're born. As we grow up, it becomes atrophied. Okay, I'm gonna say that more in the thymus, but I'm just letting you know, what does it mean? It's not constant. And sometimes these nodules, they're gonna disappear. Okay, like because a nodule is basically okay, what a nodule is. Let's do that first. There's primary nodules and there's secondary nodules. Okay, primary nodules are they have what's it called, lymphocytes, and they have no germinal center. Okay, as a definition, so it looks like dark. It's just like a dark spot. Okay, secondary nodule, they have a germinal center. Okay, which is a site of active proliferation and antibody formation that appears light. Okay, so this is a germinal center. Germinal means like when something's being formed. So it's like where the antibodies are formed, okay? There's also the mantle zone. This mantle zone is also called the corona. Do you guys know what a corona is? You guys probably know, but what's a corona? What does corona mean? Corona means crown. Okay, so coronavirus is a virus that has something that looks like a crown. Okay, so when we say the mantle zone is also called the corona zone, it means like it looks like a crown. It looks like this germinal center is wearing something. Okay, this is a mantle zone. Okay, so this is basically it. Like you just, I'm just telling you like the anatomy of them. You're gonna learn more about the physiology and how they work later on, like in immunology. But the anatomy of a lymph node. It's either primary lymph node or there's a secondary lymph node, okay? Secondary lymph nodes is where there's active proliferation, okay? So if you see these light areas, okay? Secondary. It's also called follicles, by the way, guys. So if you hear like nodules or follicles, same thing, okay? So SF means secondary follicle. If there is a secondary follicle, okay, and it has a light germinal center, it means it's an active proliferation. There's something going on inside now, like cells are being proliferated. Primary, it's just like they're there. They're finished and they're just staying there. They're chilling. Okay, they're not encapsulated. Okay, lymphatic nodules are not encapsulated. You have some lymph lobules. It's different. They have capsules and everything. These do not have capsules. So why is that important? Because if you see it in an organ or anything, and you see this like it's just a halo of dark, it's not a capsule. Then it's probably a lymph nodule or a follicle. Okay. Small lymphocytes are immature lymphocytes. Yes, immature. Germinal center contains B cells, right? Yes, it contains B cells. See, you guys probably took this at some point, but again, anatomy-wise, germinal center, it's light, it has proliferative. Okay, so immature cells. Okay, now the thymus. Remember we said what was a thymus? Was it a primary or was it a secondary organ? What was the thymus? Primary organ, exactly. Thymus is a primary organ. Okay, why? Because it's the site of maturation. Okay, does anyone know what cells mature in the thymus? We said there's a thymus and bone marrow. T cells, yes. T for thymus, T for T cells. Okay, B for bone marrow, B for B cells. This is where they got the B and T. I think. No, actually, I'm pretty sure. Okay, so it's a primary lymphatic organ, okay, and it starts to atrophy after childhood. So we're all born with a very like it's a thymus, it's a full thymus, okay. As we grow older, it becomes it starts to atrophy. So like the cells that fat cells just like go inside and cells die and everything. So this is like a full thymus and this is it being atrophied, okay. It's not as purple or like it's not as active, okay. And this is normal. This is not pathological. This is how it happens. It starts because when we're younger, we need to develop our immunity, okay? Once we develop our immunity, we have our T cells, we have everything. We don't need it anymore. So it's going to be like, bye, and it's going to disintegrate, okay? So it's an organ that has a surrounding capsule, okay? When I say capsule, guys, I'm not just saying it like, oh, capsule, fun fact. No, I'm saying it because histologically, you need to be able to tell and know it's a capsule. So here, let's zoom in a bit, okay? This is the organ. This right around it, the fibrous capsule, this is the capsule. Okay, this is why I need you to know. And thymus is a capsulated organ, which means it has a capsule. So when you see it on histology and it has like an arrow pointing there, what are you gonna say? You're gonna say it's a capsule. Okay. Okay. The capsule also it's used to divide it. Okay, so it's like think of it as an entire piece of meat. Okay, it's not going to function as one big unit. It's not a heart, okay? The thymus functions as like individual units, okay? And these units are called lobules, okay? Each lobule has something called a cortex and a medulla, okay? We're going to go into detail, but basically, thymus is a big organ, okay? The capsule is going to like invaginate or it's going to like divide it into lobules. Each lobule has a cortex, which is the outer layer of it, and has a medulla, which is the central layer, okay? The function, like we said, it's a site of T cell maturation and education. It's something called T cell education. They're released by the thymus and then they accumulate in the lymph nodes and the spleen. Okay, so they're not stored in the thymus because you can't store them there because it's gonna die. We said like thymus is not gonna be there forever. It's just like a temporary organ. So you can't store the T cells there. So they're made in the thymus, okay? They're matured and they're educated there, and then they leave and they go to the lymph nodes and the spleen. Okay, I think there was a question. T cell. Is the paracortex in the thymus or the spleen? Paracortex is in the spleen. We're gonna go into it. How are lobules different from nodules? Okay, how are lobules different from nodules? That's a very good question, okay? Lobules, they are surrounded by capsule. Nodules are not. Remember what we said here? Where is it? Oh, hello? Nodules, they are sharply defined, but they are not capsulated, okay? If they are capsulated, that means they are a lobule. And they're usually like bigger, like nodules are just like spots in there. Okay, perfect. Do they have crypts? You can call them crypts. Yes, crypt, invagination, whatever it is, it means like something that's like going inside the surface. Okay, so that's the thymus, like the anatomy of it. Now the cells of the thymus. Okay, this is important. Okay, the lobule is a circled part, right? Circled part. Yes, it is a circled part. Now the cells of the thymus, okay? We said it's primarily T cells. Okay, this is where they mature. Okay, they are active from from the medulla. They are more active. Wait, I'm sorry. From the cortex to the medulla, they become less active. So that means they are most active in the cortex. Okay, this makes sense from the picture. Okay, sometimes you get confused from like reading, like I just did. So look at the picture. It's darker, means it's more active. Okay, so they're active in the cortex, and as they go into the medulla, they become less active. Okay. Now, there's something called the thymus epithelial cells. Remember when we talked about the supporting network, and we said that the supporting network is either reticular cells or reticular fibers and cells? Thymus is an organ that only has reticular cells. Okay, does not have reticular fiber. So when you look at this like picture right here, you don't see any fibers, right? There's no fibers here. There's no fibers. These are like blood vessels. They're not fibers because there's no fibers. Okay, it's most likely a thymus. Okay. The reticular cells inside the thymus are called thymus epithelial cells or reticular epithelial cells, whatever it is. Any combination of the word thymus, epithelial, reticular, and cells, that's the reticular cells of the thymus. Okay. The network is maintained by the protrusion. So don't worry, like because they don't have fibers means they're not gonna have support. No, they're still gonna have support. It's just their support are made from their protrusions, like a dendrite. Okay, not from the fibers. I think there was a question. So immature T cells are less active. Yes, less active in the body. Like metabolically, they're very active because they're being matured, but in the body, they don't have a function yet. They're still growing up, you know. Now, B cells can be found in the thymus. Okay, so yes, we said it is the main location for T cells, but you can also find some B cells in like follicles or nodules. Okay. Now that we said there's a cortex and there's a medulla. The cortex is dense and it has immature T cells. Okay. The medulla is lighter and it has mature T cells. Okay. So I can see, I think, where the confusion came from. Active meta when it's active, okay, if it's mature, it's not doing anything. Khalas, like it did its job. So it's just gonna stay there. So that's why you said from the cortex to the medulla they become less active. You know that I mean they're not maturing. So in the medulla, they're all mature. Khalas, like they're lighter, so there's no metabolic activity going on. They're not doing anything because khalas, they reach their final stage of life. Okay. The ones in the cortex, they're immature and they're active because they're actively growing. You still need to feed them. Think about it as like a very old person and like a toddler, okay? A toddler is always jumping around, they're eating, they're cooking, they're whatever it is. An older person, no, they're more chill, خلاص, they're sitting on their chair, they're reading a book, whatever it is, okay? This is what I mean, and no, they're more active in the cortex because they're immature, okay? They're immature, so they're active. Mature means خلاص, they're not active. They did what they needed to do. Is it necessary that the dark part is always active and the light part is less active? Although in lymph node, it's the opposite. Good question. We're gonna come to it when we do the lymph nodes. I get what you mean, but let's see in the lymph nodes and let's see if you understand it more. Okay. So yeah, this is a cortex. It's dense. It's much more darker because it has immature T cells. The medulla is lighter, has T cells, and there's something called Hassel's corpuscles. Okay. Let's go to it now. The thymus capsule, like I said, it's capsulated organ. Okay, so it's a fibrous connective tissue, capsule, which it has tuberculae or invaginations into the organ itself. So this is what divides it. So you see a picture like this, guys, this was the same picture that came to us in the exam. Okay, it's an organ. It has a fibrous capsule. You can see right here. It's surrounded by capsule. Okay, it's divided into lobules. Okay, it has an inner medulla and it has an outer cortex. The cortex is lighter and the medulla is the cortex is darker and the medulla is lighter. This is most likely a thymus. Okay, so I was able to identify all the aspects. I was like, it's a fiber, it's this and this and that, but I couldn't say it's a thymus because I forgot the name. So where are the blood vessels? They're found usually in the tubercula or the capsule, yani inside. When the capsule goes inside, it carries with it the blood vessels and everything. Okay. The lobules, like we said, and it has a cortex, the medulla, and corpuscles. Okay, how can you tell which is which? So how can you tell this? I'm looking at a cortex and I'm looking at a medulla. The main thing that we said in the cortex is usually darker and the medulla is lighter. But what if you have just like this? What if this picture came to you? Are you able to tell it's light or dark? Yani if you don't have anything to compare it to, you usually can't tell. Okay, unless because I can see both of them beside each other, I can see this is darker and this is lighter. But if I have just one picture, how can you see? The one thing that's like very characteristic, very, very characteristic of a medulla is the what's it called? the corpuscles, the Hassel corpuscles, okay? Right here, this one. It's just an aggregation of some cells. The function of it is not actually completely known. It's just like it exists there. It just is chilling there, okay? So if you have corpuscles, okay, right there, you see a cell, it's like very clumpy and it's just like a big giant cell. This is Hassel's corpuscles, okay? So this section is medullary section, okay? This one is probably not because it doesn't have any, I don't see any corpuscles philic. It's so basophilic, it's most likely the cortex, okay? So this is how you can differentiate. And again, like we said, it has reticular epithelial mesh or reticular epithelial thymus epithelium reticular means no fibers. You did not hear me say fibers at all. So no fibers. So it's cells, it's reticular cells, reticular reticular cells. Okay. This is another picture. Okay. The medulla, the cortex, and this is a corpuscle. So this is probably a what? This is probably a piece of the medulla. Okay. This is another part of the supporting network, the blood-thymus barrier. Okay, we have a lot of barriers in our body. We said there's a blood-brain barrier. There's something else called the blood-thymus barrier, okay? Which is what prevents T cells, okay, from leaving into the blood or the blood going to the T cells. Why? Because the thymus inside it, there's still immature cells. You don't want them to be exposed to any antigens yet. They're still cooking. You know, leave them, let them cook. So while they're still like maturing, you don't want anything to come near them, okay? Because if it did, it can like ruin the entire process, okay? So you want to leave them by themselves. That's why you need to have a blood-thymus barrier, okay? Now, the important thing from this slide that you need to know is that the components of the blood-thymus barrier. What does it have? Capillary endothelium. It has perivascular connective tissue occupied by macrophages, or basically macrophages around the vessel, and it has type 1 epithelial reticular cells. A lot of big words. You only need to memorize it. Okay. Capillary endothelium, it has capillaries. Okay. Capillary endothelium, very easy. There is macrophages. See, this is a macrophage. Macrophage. All these are macrophages. Okay. This is the inside of the thymus. These are the developing T cells. This is what we're trying to protect. You don't want anything to come near them. Okay. So the blood-thymus barrier, the capillary wall. Okay. The perivascular connective tissue, which has macrophages, and the epithelial reticular connective tissue layer. Okay, big words, like I said, just memorize it. It has macrophages, it has capillary endothelium, it has epithelial reticular cells. Okay, does that make sense? Is that easy? I know it's a bit hard because of the names, but as a concept, do you guys get it? Yes. Okay, perfect. How can you differentiate between thymocytes and reticular cells? Good question. There was a picture, I think, right here. Okay, see these cells? These are probably the reticular cells. These dark basophilic cells are probably the thymus cells, because what are the thymocytes or the thymus cells? They're the T cells, okay? So these are probably the T cells, and these are the reticular fibers, okay? Reticular fiber cells, like we showed it before in the, what's it called? Supporting network. It's a cell that has a nucleus and it's a bit elongated, that these are probably the reticular cells, because anything else around it is just thymus cells. And these are the corpuscles, okay? Okay. Yeah, you skipped a few questions though. I skipped a few questions. Nodules are absent in the thymus. So aren't follicles and nodules the same thing? Follicles and nodules. Okay, good question. Why did I say nodules are absent in the thymus? Because thymus is not the location for B cell maturation, okay? Here, let's go back to this cell, lymphatic nodules. What were the nodules? The nodules were, there's a germinal center and there's the corona and everything. But the main thing about the lymphatic nodules is where the B cells are made or created, okay? So when I say there is no nodules in the thymus, it means that there's no B cell activation, okay? It has something else. It has their own lobules, okay? Lobules because it has the connective tissue and has a medulla and a cortex, okay? It's not, so like, okay, what's the other organ that has nodules and stuff? Lymph nodes. Lymph nodes, they have a lot of nodules, okay? They're all made of nodules, and it has like the primary and secondary. But a thymus doesn't have, like, okay, look at the thymus. There's no nodules, okay? It's just one big lobule that's separated into a cortex and a medulla. So that's what I mean when I say there's no nodules. And the thymus has no nodules because there's no B cells maturating everywhere. We said there's B cells somewhere scattered, but yani it's not the normal. But this is what I mean, okay? Does that make sense? What is the function of a trabecula? Function of a trabecula in the thymus, that it separates it. Okay, we're gonna go into more detail on the trabecula and everything in the spleen, because this is where it actually like has a function. But the trabecula means like an invagination, and it has the capsule with it, and with it comes some blood vessels. So the main function, you can say, of the trabecula is that it contains the blood vessels and the nerves and everything that's the like support the organ. Okay, so trabecula is like the crypt itself is like the little invagination. It's surrounded by the capsule, because we said it's the capsule, the one that like separates them. They separate them in the space. So usually a capsule inside there has some blood vessels. Okay, you're gonna understand it more in the spleen. But for now, did you get it? Is it clear? Perfect. Okay. You finish this one, support and network. Okay. Now we finish with the thymus. Okay. I know it's a bit confusing, guys, the thymus. That's why like I spent so much time in it in first year. But if you go back to these slides and just like look at the pictures very well, and you recognize, okay, so let me ask you a pop question right now. If you got this exact picture in the slide, these two pictures in the slides, okay, I'm gonna ask you um what divides the lobules of this specific organ and what thymus, and what area is this? Okay, so what divides it? What's the thing? Trabecula or the capsule? Better answer would be the capsule and a trabecula. And what is this? Hasselback. Yes, it's a Hassel's corpuscle. But what's the area? What's it called? Medulla. Exactly. See, this is how it could come to you in the question. Okay, it could be like, what's the invagination is made of? It has capsules inside. Okay, what's the difference between a cortex and a medulla? And the cortex has the immature T cells, and the medulla has the mature T cells. Cortex is darker, medulla is lighter. Okay, so if you're still confused, it's a heavy topic. I know the thymus. Just go back to it. But still, know that like the things that differentiate everyone from each other. So like Hassel's corpuscle, very, very important. Okay, most likely they're gonna give you a question, and it has a Hassel's corpuscle, and it's gonna be like, what is this? And where is it found? It's gonna be found in the medulla of the thymus. Okay, what's the white part called? White part. What white part? Where do you see white? Do you mean these? The lines, Danny? Yes, these are this is the invaginations or the tubercula. Okay, it's not, it's like the lack of yani, this is a big thymus. Okay, you cut it inside it to divide it. These divisions, these are the trabecula, and like what makes it is a capsule. Okay, okay, guys, what I mean with the capsule makes it, because while it's developing, like while the thymus is developing, it's like, think of it as a big blob. It's just like a blob of organ developing. Okay, around it you have the fibrous capsule. Okay, when it's developing, these capsules is gonna go inside. It's gonna make crypts, trabecula. It's same name, different same thing, different names. It's gonna make crypts. It's gonna make trabecula, whatever it is. Yani with the capsule moving with it. This is what divides it. So each lobule is divided by its own capsule, because while it was separating, the capsule went with it. Did you get it? White parts are crypts. Yes, they're called crypts, trabecula. CT, yes, CT. It's dense connective tissue. That's what it's a dense fibrous connective tissue. That's the capsule. How do you tell the difference between germinal centers and the cortex / medulla? Good question. You're not going to find germinal centers in a thymus. Okay, when you do find them, it's going to be in like a lymph node. Let's go back here. Yeah. See here, مثلا, these are like, this is how it looks like. Okay, see how in the thymus it was like lobules, and it has cortex, and it has a medulla. It's the entire organ. It's like, the difference is mainly in the size. If it's a big organ, a piece of an organ, it's like separated into a cortex and a medulla. This is the cortex and medulla of a thymus. Okay, if it's a piece of an organ, and inside it has little nodules, and each nodule looks different, like one of them is dark, one of them has a light center, this is a nodule. Did you get it? Too many questions, عادي. But regular or irregular CT, the crypts I mean. Regular or irregular. Let's see what was written here. One second, guys. And so I forgot, but it was dense connective tissue. Okay, all capsules are usually dense, fibrous. Okay, so if you wrote that, it's fine. I don't think they're gonna ask if they were regular or irregular. So you can repeat the lobules versus the nodules part. Yeah, sure. Okay. For the question, Tara, for the irregular, I'll look for it and I'll add it to the slides by then because I don't think I know it was written somewhere. So for the thing, for the lobules and the nodules, okay? Lobules means a lobe. What does a lobe mean? You guys know what a lobe is? Lobe means a part of an organ, okay? It's an organ that was divided into another part, okay? So a lobule is surrounded by a capsule, okay? Right here, it's written right here, and has a surrounding capsule that divides it into lobules, okay? So that's a lobule. A lobule is a piece of an organ. An organ was divided into a couple of pieces. So this is a lobule, this is a lobule. This, wherever you see connective tissue, it's a lobule. So this is a lobule. This is a lobule. This is a lobule. And every lobule has its own cortex and medulla. So let's take this, for example. See how this one already has its own connective tissue? Like this one probably passes through here. It has a darker outside and a lighter center. Okay, so this is what I mean by lobules and what lobules are. Nodules, let's go back to the nodules. Hello. Okay, nodules here. See how they don't have a capsule. This is the most important for if there's anything that you don't understand and you want to understand something, understand that lobules are surrounded by capsule. This is what makes them a lobule. It has capsule around them, okay? Nodules do not. Nodules are not encapsulated. See, they're not encapsulated. They're just by themselves. They identify themselves by like the halo of a color. It's like difference in gradients, okay? So nodules, okay, good, perfect, خلاص. Nodules, like I said, not capsulated, and it has some of them can either be primary or secondary, okay? Lobule is surrounded by capsule. خلاص. Okay. Okay, we finished the thymus, which was the heaviest part of this lecture, so the rest is very easy. Tonsils, okay, tonsils. What are tonsils? You guys know what tonsil is. Did anyone ever get a tonsillitis before? It's a horrible, horrible thing. Tonsillitis, Lewis, yes, exactly, Lewis. These are the tonsils. The tonsils are a part of the lymph system. We said the lymphatic system is made of organs and tissue. So one of the organs of the lymphatic system are tonsils, okay? They're immune organs, okay? So where are the tonsils found? They're found in an area called Waldeyer's ring. Okay, well, Waldeyer's ring is like an area surrounding, it's like from the tongue, like right under the tongue, here over the nasopharynx, and like it's like if you take your throat and like you take this part, you wrap it here, this entire part like surrounding your esophagus, this is called Waldeyer's ring. This is where the tonsils are found, okay? So basically know that tonsils are not going to be found in your fingers. No, tonsils are only found in like the entrance of your throat, is the epithelial lining of your throat. Right underneath the epithelial lining are the tonsils. Okay, and the area itself is called Waldeyer's ring. Just know that. So Waldeyer's ring is an area at the entrance of your throat that contains the tonsils. Okay. So what are the tonsils? There's three main groups. Okay, these are what you need to know. There's palatine tonsils, there's the pharyngeal tonsils, and there are the lingual tonsils. Okay. Now they're very easy to know from their names. خلاص. Palatine tonsils. Okay. Where are these? These are the palatine tonsils. Okay. Right here. So this is your this is the hard palate, this is the soft palate. Right underneath it is the palatine tonsil. Okay. The pharyngeal tonsil from the name pharynx in the nasopharynx. Okay, this is a nasopharynx. Where is it? Pharyngeal tonsil. Nasopharynx. You see it? So like I said, it's the entrance of your throat. So your throat composes of the esophagus and the trachea. So this entire entrance here, this is the Waldeyer's ring. So pharyngeal tonsils, okay? This is the palatine tonsil. And then you have the lingual tonsil. Where's the lingual? Lingual right here. It's like at the back of the tongue, like the last part of the tongue. That's where they're usually found. Okay, so you just need to know what the difference between them is. So palatine tonsils, the epithelial of it is covered by non-keratinized stratified squamous epithelium. Big words. So you guys took this before in the beginning, okay? Easy. Non-keratinized, no keratin. So in the picture right here, شوفوا how is no keratin. This is cell, okay? On the other hand, pharyngeal, that is covered by pseudostratified epithelium and it has keratin, okay? So where is it? Here. شوف this like white part, this is the keratin. So pharyngeal are keratinized, palatine are not, okay? So if you see it is non-keratinized, stratified squamous epithelium. Stratified means on top of each other. Squamous means the flat cells, so stratified squamous on top of each other. They form, they have invaginations or crypts, okay? So this picture right here, this is probably a palatine tonsil. Okay, so it has invaginations. This is the epithelium of it. Okay, no, I'm kidding. Yeah, here, scripts and stuff like that. It doesn't have any keratin. Okay. So pharyngeal tonsils are found in the nasopharynx. If it's found in the nasopharynx, it's surrounded by pseudostratified respiratory epithelium. Okay, so if you get confused, what's pseudostratified? Remember the respiratory epithelium. Respiratory epithelium is pseudostratified epithelium, and that's a respiratory epithelium. Okay, so pharyngeal right here, pseudostratified columnar over here. This is how it looks like. Palatine is squamous. Okay. The other difference is lingual tonsils. Okay, they are also under the squamous epithelium, the base of the tongue, has crypts and receive ducts of saliva. Okay, so here, because they're so different, like similar to each other, what's the difference between palatine and pharyngeal? Palatine has non-keratinized stratified squamous. Pharyngeal has respiratory epithelium. Okay, lingual has salivary glands in them. So this is how you can identify them. If you see a picture with salivary glands, so for example, this one. Does this one have salivary glands? I can't tell. If it has salivary glands, okay, usually they're gonna tell you it's lingual tonsil. If they said the location is under the tongue, it's lingual tonsil, okay? Pharyngeal tonsils, if they said it's found in nasopharynx, it's pharyngeal. If they have respiratory epithelium, it's pharyngeal. If palatine, if it's non-keratinized stratified squamous epithelium, it's palatine. Okay? Did you get it? Is it clear? Yes, perfect. Okay. Yeah, so the most important part, صراحة, from here, and no, just know how you can identify each of them from the location, from the epithelium, and if it has saliva, then it's lingual. As again, picture, this is a pharyngeal, this is palatine, and I'm gonna check. I think this is a lingual tonsils. Okay, I'm gonna label it too after the pal, inshallah. Okay. Now the lymph nodes. Okay, lymph nodes are they're the big guns. Okay, you have lymph nodes everywhere. Everywhere you have lymph nodes. Sahwi said tonsils are only found in the Walden's ring. Lymph nodes are found everywhere. Okay, they're some important lymph nodes that you guys probably took. The ones right here, the ones right here, the subclavian, whatever, the ones for breast cancer. So lymph nodes are very, very important. So they have some important aspects of it is the vessels and the hilum of the lymph nodes, okay, and the capsule. They are capsulated. What other organ was capsulated? What did we say was capsulated? Thymus, exactly. Thymus was capsulated. Lymph nodes are also capsulated. So remember that if you're looking at a histological picture of lymph nodes and it tells you like what's an identifiable feature, say it's capsulated because lymph nodes are capsulated. Okay. Now vessels. The important thing from lymph nodes, guys, is that lymph nodes, it's what filters blood, okay? Where blood is filtered or how it's filtered is in the lymph nodes and the spleen. I'm saying lymph nodes are like the tiny stop stations. Like they take the blood and they're like, okay, let me see if you have anything bad, and then you can go on your way, okay? So the vessels, you have afferent and efferent lymphatic vessels, okay? Afferent means they go towards the node. Efferent, they go away from the node. Okay, it's the same thing with the nerves, okay? If you took that, efferent nerves and afferent nerves. Afferent means they're going to the nodule. Efferent means they're going away from the nodule, okay? And so they also have valves. Again, any lymphatic vessel, they have valves. The hilum. Now, the hilum of a lymph node is this. This, like, see how it's like convex over here? It's like, it has surface like this. The second its surface starts to go in, being concave, this is the hilum. Hilum means the entrance to the lymph node, okay? Why is it important? Because this is where the lymphatic vessels, this is where the artery, veins, and nerves, this is where they penetrate the organ, okay? This is what a hilum is, basically. Now, it has a capsule. We said again, it's collagen fibers, elastic fibers, contains vessels and everything, same as any other capsule. Okay. Now, this is the idea of the nodules. We come back to it. Okay, so the stroma of the lymph nodes is reticular fibers, type 3 collagen. Okay, a thymus, what did it have? Did it have reticular fibers or reticular cells? Cells or fibers? Cells, exactly. So don't forget, if it give you a thymus, okay, and it tells you what's the arrangement or what's the surrounding or supporting like substance, anything along these lines, say it's reticular cells, okay? Lymph nodes are reticular fibers, okay? So here are the reticular fibers. There they are. They're like a meshwork, they're between cells, okay? We didn't have any of that in the thymus. Remember, thymus were just like cells. There's a lot of cells, that's it. So the regions in the lymph nodes, okay? Now each lymph node has its own, it's like an entire yeah, it's an entire thing, okay? So it has an outer cortex. This is the outer cortex. Whenever I say cortex, it means the outer layer. So this is a cortex, okay? There is paracortex, which is right in between this P thing. This is a paracortex, and it has a medulla. Someone mentioned it before, and does thymus have paracortex? Thymus does not have a paracortex. It only have a cortex and a medulla. Lymph node has cortex, paracortex, medulla. Okay? I think that's a question. We have both in lymph node, right? Reticular fibers. Yes, yes. Whenever you have reticular fibers, you're gonna have cells with them because where are they gonna come from? Okay, reticular cells are the ones that release the fibers. Okay. You get the arrangement now? It's not the same as thymus node. It's divided into lobules. No, each lymph node is by itself like an entire thing. It has an outer cortex, has a paracortex, and it has a medulla. Okay. Now, remember the nodules, the primary and the secondary. Where are they found? They're found in the cortex. Okay, so nodules are only found in the cortex. Once you go to the paracortex, it does not have nodules, and doesn't also have nodules in the medulla. Okay. So again, you see all these nodules are all in the cortex, all in the cortex. And this is still it's because it's dark, it's still a cortex, but this is why it has like nodules. Again, paracortex, there is no nodules. Okay, and then the medulla. Okay, now sinuses. What is a lymph sinus? Okay, it's a structure that carries a lymph fluid from the efferent to the afferent. Okay, so we said, and we have the hilum. The hilum is where everything goes in. Okay, so hilum goes arteries, veins, vessels, everything goes to the hilum. Okay, now the sinus inside it, this is here. The sinus is what carries it from the efferent and afferent. Okay, don't be confused. Okay, let's zoom into here a bit. This is the best picture to explain. This is the lymph node. Okay, the flow of the lymph. Okay, afferent, it's going in. This is the afferent lymph vessels. It's going in. Okay, it's gonna go inside the trabecula. Okay, which divides it. Right here, divides it over there. It's gonna go into the medulla. Okay, and then this trabecular sinuses. The trabecular sinuses is basically where it moves in. So it goes from the afferent lymph vessel to the trabecular sinus, to the medulla, and then to the efferent. So this is like the arrangement, how it goes. I think it was in a slide. Yeah, right here. This is a very, very, very clear picture, okay? You have the afferent vessel, okay? The afferent vessel goes in. This is the just from the blood to the lymph. Basically, let's explain this entire diagram, okay? This is a trabecular sinus. This is the vessel itself. This is the lymph vessel, okay? This is where it's leaving. So again, we have the cortex, paracortex, and medulla. There's something in the paracortex called the HEV, which were called where is it? HEVs. Yeah. High endothelial venules. Okay. High endothelial venules, they are the site of the entrance of the circulating lymphocyte. What does that mean? The lymphocytes, where they come in and out, they come in through the HEVs. This is what's important. Okay. HEVs are the site for the excavation of the endothelial venules. Okay. Where were we? Yeah. Okay. Yes, sinuses. Okay. So macrophages are present around the sinuses, okay? Because the sinuses are the ones that contain the fluid. So guys, don't be confused. Again, there's a lot of things going into the lymph node. That's why we have different names. So the vessels are not sinuses. What's between like the vessels and the lymph node itself is a sinus, okay? The sinus contains lymph fluid. The vessels contain blood. Okay, it's easy. The vessels, there's an afferent vessel going into the lymph node, and there's an efferent vessel. These are only carrying lymph, okay? No blood. So this is what a sinus is. A sinus carries only lymph. But vessels, they carry blood as they always been. Now, macrophages are found around the sinuses. Why? Because the entire function of a lymph node is immunity. So you need macrophages. So they're found around the sinuses, okay? Now, the afferent, it drains into the subcapsular sinus, okay? So it first starts a subcapsular sinus by endothelia, and then it loses into the lining, and then it goes to macrophage lining. What that means? It means I have a pen right here. Again, this pen is like going through it a lot. It's gonna go, it's like the sinus is moving. Okay, as it's moving, it's covered by endothelium, Adi. Once it goes inside the subcapsular like space, it loses its endothelium and it becomes instead covered by macrophages. Okay. Yeah. So where was the picture that we were explaining in? Here. This again, how it moves in, like it loses its like lining and it has more macrophages. This is the subcapsular space. Subcapsular here, right here, capsular. Okay. This is the afferent. It goes inside a subcapsular sinus. Okay. So it goes in first as an afferent vessel. It turns into a subcapsular sinus. Okay. Then it turns into a trabecular sinus. Okay. And what was the last one? Medullary sinus. Okay, so sinus, again, carries lymph nodes. First one goes from the afferent. Afferent turns into the subcapsular sinus. Then that turns into a trabecular sinus, which turns into a medullary sinus. Okay, it's just the stages where lymph nodes go through. This is location, just talking about the location. Again, this picture was in the slide, so just know it. And this is a capsule. Okay, right here, capsule. And this is the subcapsular sinus. Okay. Now the vascular supply. Is there any questions first? Is everything good till now, guys? I know it's a bit confusing, صراحة. There's a lot of information, but like the things I keep repeating are the ones that are important. You know, like what the sinuses are called and like what they carry. Sinuses carry lymph. Okay. Yes. Okay. Perfect. Okay, vascular supply. Like we said, there's an artery nerve and a vein. Okay, artery goes in, veins go out. Okay, and also the afferent vessels goes out. Okay, here they're going into they're going into the medulla and then to the cortex and to the cortex. Okay, so the arteries and veins, they pass through the higher lymph first, give up branches. In the cortex, they form anastomosing loops. You know what anastomosis means? Does anyone know what anastomose means? Anastomosis. No. Okay. It's fine. Anastomosis means when two vessels, okay, so usually two capillaries or two vessels, like it's vein, they come together and they make like a meshwork. Okay, this is found in so many spaces. Like when you take the what's it called? The vessel, the blood supply of so many organs have anastomosis. Okay, anastomosis basically means a meshwork of fibers. Okay, so that's what anastomosis means. So the anastomosis of the vessels for the lymph nodes, they occur in the cortex. So see how like they're like fingers together? This is the anastomosis. It's found in the cortex. Okay. Now remember we said the post-capillary HEVs, these are found in the pericortex. Okay. So cortex has a what? What did the cortex contain? It contained, we said, anastomosis, but also had something else. Remember this is found in the cortex of lymph nodes. What is found in the cortex of lymph nodules? Yes, yes. So cortex has nodules. Again, this is a distinguishing feature. If they ask you where are nodules found, they are found in the cortex. Now, paracortex. What is found in the paracortex? What we just mentioned was found in the paracortex. Paracortex. The HEVs, okay, or the high here. The high endothelial venues, HEVs, are found in the paracortex. Okay, so here, cortex has nodules. Okay, paracortex has the specialized postcapillary venules or the HEVs. Okay, these are again the sites and where the lymphocytes they go in and out. Okay, so again, cortex and the nodules, the primary or secondary for the B cells. Paracortex at home between the medulla and the superficial cortex. It contains most of the T cells. If there are T cells in the lymph node, this is where they are. So paracortex contains T cells and they contain the HEVs. Okay? Okay. Is there a question? So LIMS also comes from yes. Okay, basically think of HEV as the ending of the endothelial vessel. So you know how there's an artery and then goes arterial and there's a capillary? The capillary for vessels is the HEV. Did you get it? So it has lymph, yes. So HEV, again, it's lined by cuboidal or columnar endothelial cells because it's like a duct. So here, columnar, this is cuboidal cells, okay? Here they are. You're gonna find it like in like we said in the paracortex. This is the paracortex, okay? In the middle of it, you see a vessel that contains fluid inside. This is probably an HEV, okay? 90% of the lymphocytes enter the lymph node through the HEV. So lymphocytes, they enter and exit the lymph node through the HEV. Okay, this is basically it. The medulla now is it basically has something called cords. Okay, cords are what are cords? This is a cord. MC is a medullary cord. This is a medullary cord. Okay, and it also has medullary sinuses, so medullary sinus, okay? So the medulla is basically a collection of medullary cords and medullary sinuses, okay? One of them, like, this is a cord, this is a sinus, this is a cord, this is a sinus, okay? This is where they are. So the lymphocytes, the B cells and macrophages, the dendritic cells, everything, they're found in the cords. So the cords are where the cells are. So where the cells are located are in the cords, okay? The sinuses, they contain, again, the lymph itself, the fluid, and macrophages, because we want to filter it. Okay, the lymphocytes migrate from the cortex to the medulla to the efferent vessels to leave the lymph node. Like, this is the path, basically. And it goes from the efferent arteriole to the sinus. The sinus keeps going into from the subcapsular to the trabecular to the medullary sinus. This is what we're talking about, the medullary sinus, okay? And then it goes out through the efferent lymphatic vessel. Do you get it? This is it for the lymph nodes, Yani. Is it clear, guys? Is everything... Can you give a quick summary? Yes, of course. Okay, quick summary for the lymph nodes. Let's use this picture here because it has a lot of stuff. Actually, where was the schematic one? Okay, this is a good picture. Basically, lymph nodes. A lymph node is an organ by itself. Okay, so it has something. It has like a round surface and it has this concave surface, which is called a hilum. Okay, what's a hilum? A hilum is where the lymphatic vessels and the blood vessels and the veins, they enter and go. Okay, so we said we have the lymphatic vessels are two things. There's the afferent and there's the efferent. Afferent, it's going into the lymph node. So it's the afferent is carrying lymph, okay, lymph fluid, yani, going into the lymph node. It gets filtered and everything, and it leaves through the efferent. Okay, here again, this is how you see it. It's going into here. Meshi. Now, when it goes inside, it has a trabecula. This is where the trabecula is actually important. The one for the thymus was more of like a anatomical trabecula, or like a it was the one divided into lobules. Okay, this one is actually functional. Why are they needed? Because this is where sinuses move. Like, you see how they move? They move into the trabecula. So the trabecula itself is acting as a vessel. You get it? So again, the afferent lymph vessel, it carries the lymph. It goes into the sinuses. So the sinuses has three parts. It has first the subcapsular, and then this is capsular right under the capsule, then trabecular, and then medullary. Okay, trabecular sinus, medullary sinus. Okay, the medullary sinuses, like we said, the entire medulla. Show like how the medulla looks like. It's like this is a sinus, and this is a cord. This is a sinus. This is a cord, because this is where like everything in it gets filtered. There's a lot of macrophages. There's a lot of B cells. There's everything. This is where it gets filtered. Okay, so once it enters the medullary sinus, okay, it's going. Where is it? Yeah, once it goes into the medullary sinus, خلاص, it's gonna go into the efferent lymphatic vessels. Okay, so it goes in from the afferent, goes to the trabecular sinuses, all the sinuses, into the medulla, which has cords and sinuses to medullary sinus, and then شوفوا there's blood vessels and everything. The blood, like it goes in and out, the interstitial fluid goes in and out, and then it leaves through the efferent. Did I miss anything? Yeah, again, the HEV are like the capillaries of the lymphatic vessels. So it's like the ending of it, the very ending. And this is where the lymphocytes themselves, like B cells and T cells, they enter and exit. Okay? And it's found in the paracortex. Okay? The identifying features. If you see a nodule, it's in the cortex. If you see an HEV, it's in the paracortex. Okay? If you see medullary sinus and it's just like sinus and cord, this is the medulla. I think this is like it. Is that good? Do you need me to do more? Clear, I think. Perfect. Okay. Okay, we're done with the everything. We have the last part, which is a spleen. Okay, so bear with me, guys. Just last push. What's a septi?
Sorry, Jenna, can I just interrupt you for a second?
Yeah, sure.
Everyone, for the sake of time, can you please keep your questions to the end so that we can end the recording and it won't be as long for the people watching? Thank you. Jenna, can you just like answer the questions at the end? So
Sure,
like,
sure.
okay, perfect. Cuz, thank you.
I got yelled at. Okay. So let's finish the last part very quickly. The spleen, okay, guys, the spleen is the largest lymphatic organ. what we say the lymphatic organs were, there was a thymus, there's lymph nodes, the largest one is the spleen, okay? So it has, what does it do? In the veins and arteries, the blood goes inside, and this is where the blood gets filtered. So if you ever saw in a movie, or like, I don't know if you read about it before, if a spleen gets injured, the person will bleed so much, like it bleeds a lot, because all the blood is in it. Like the blood goes through the spleen, gets filtered, and goes back out, okay? So many blood, and it has no afferent lymphatics, okay? So it's not like a lymph node and you have an afferent vessel that goes in and goes out. No, it has no afferent. It just like, it leaves back to the veins, okay? Now it contains special vascular spaces or reticular cells, and it has a lot of like macrophages and a lot of dendritic cells. We're going to talk about it now. And this basically monitors the blood immunologically. Okay, so when you think about an organ, there's blood filtering, blood reservoir, the spleen. This is the spleen. Okay. Now the capsule, it's a capsulated organ. Again, what was capsulated? We said the thymus was capsulated, the lymph nodes were capsulated, and also the spleen is capsulated. Okay, so it's capsulated by a dense connective tissue. Okay, and also has, again, trabecula and everything. Now the trabecula here, it doesn't really have a function anatomically. It doesn't like separate to anything. It's just like it's just there. Okay. Now the trabecula here, it contains myofibroblasts and contractions. Oh, never mind. Yeah, it discharges stored blood. So here it says it's a reservoir, so where is it? Reservoir, okay? Usually the blood is stored within like the trabecula. Once there's like low blood or anything, they contract and just pushes blood. It's not like that important صراحة, it's just like it was mentioned, okay? It also has a hilum. Remember what had a hilum? A lymph node had a hilum, okay? So the spleen also has a hilum, okay? It's the passage of the splenic artery, splenic vein, nerves, and lymphatic vessels. We said there's no afferent lymphatic vessels, but there is an efferent, okay? Now the pulp, the main parenchyma of the spleen. Parenchyma is like the functional unit, okay? Now the entire spleen is functional. It's not like مثلا إنه a thymus, إنه like each lobule has, no, the entire spleen itself is has a function of the whole blood filtration, okay? So this is stroma. Stroma means يعني the everything of the spleen, like the surrounding everything. The parenchyma is the functional aspect of the spleen, okay? It has something called white pulp and it has something called red pulp, okay? So white pulp is on like 20%. What's white pulp? Don't let the name fool you guys. It's not actually white. It's like blue. This is the white pulp. This is the white pulp. Okay, I don't know why they named it white. Maybe another stain, it's white. But this is a white pulp. Everything else around it, a red pulp. Okay? So white pulp is masses of the spleen, consists of lymphoid tissues, and it follows the arteries. So like it's, you see how in the center of it is arteries. This is the the white pulp. Red pulp is the one that's like more abundant. It's like everything there, and it has sinusoids and splenic cords. Remember how in the the lymph node there was cords and the medullary cords and medullary sinuses? The spleen has the same, just called a splenic cord and splenic sinus. Okay, this is where the lymph node passes by. Whenever you hear sinus, it's lymph. Now, the white pulp. What's a white pulp? White pulp, okay. Basically, the artery, when it goes inside the spleen, it's going to move in and then it's going to exit through the trabecula. When it exits through the trabecula, okay, it's going to enter the white pulp. What's it going to enter as? It's going to enter as something called central arterial. So again, trabecular artery. Once it goes into the spleen, now it's called a trabecular artery. It's the same as a trabecular sinus, remember, but now it's an artery, it carries blood. It goes inside the spleen, now it's a trabecular artery. If something branches from it and now it goes to the white pulp first, now this is a central arterial, okay? Now it has something that wraps around it. It's called periarteriolar lymphoid sheath, or PALS, okay? So a PAL, see this white halo thing around it? This is called periarteriolar lymphoid sheath, or it's mainly composed of T cells, okay? Some macrophages and dendrites too. But again, this is the white pulp. So when there is a vessel that leaves the trabecula, see, this is the trabecula. Now it's not in a trabecula anymore. It's inside a PAL. So it leaves from a trabecula to a PAL. This is the first part of the blood moving inside the spleen. Okay. Now the red pulp, once it leaves the, this is the ending of the white pulp. Okay. So this is the PAL. Once it leaves it, it loses its sheath. It loses the PAL. Okay. And then it turns into a what's it called? Peninsular artery. I have a problem with pronouncing it, but it's peninsular arterial. This is what it turns into. So again, first it's in the trabecula. It's a trabecular artery. It leaves. It becomes a central arterial. It doesn't have a trabecula anymore. It has a PAL around it, and now it's in the white pulp. Okay. Once it leaves the white pulp, now it's called a penicillar artery. Penicillar arteriole. Okay. This is the red pulp. You see this entire thing. It's composed entirely of cords and splenic sinusoids. So this is a cord. This is a sinusoid. This is a cord. This is a sinusoid. The white part that has lymph in it, is a sinusoid. The one that has cells and everything. This is a cord. Okay. Now we see these in the schematic. It has like these openings. These are the sinusoids, like the inside of it is a sinusoid. It has something called stave cells. Okay, stave cells is like a filter. It's the filter. Okay, it's the one that decides what goes in back to the blood or like to the lymph. Okay. Okay. Now this is a circulation overall. Okay, of a spleen. This is like the whole spleen. If you understand anything about spleen, this is what you need to understand. The blood vessel, the artery, the trabecular artery is gonna go into the spleen. Okay, inside the trabecula. Once it leaves the trabecula, it becomes it's called now a central arteriole. Okay, and it's covered by a pal. Okay, it's in the white pulp, which is mostly B cells. Remember, there's germinal center and everything. This is a white pulp. This is the white pulp. Okay, so it's in the white pulp. It's there. Now, when it leaves the white pulp, now it's called a penicillar arteriole. Okay, now these pericillar arteriole. The pericillar arteriole, this is where the blood's gonna leave. So there's two ways it's gonna leave. It's in your slides, بصراحة it's not that high yield, so I'm gonna explain it to you just by talking. I'll include in the slides بعدين. There's something called a closed circuit or an open circulation. Open circuit circulation, closed circuit circulation. This is a closed circulation. إنه على طول the penicillar artery, it goes into the sinusoid and it's like filtered inside. The open circulation is when it leaves and it like diffuses into the sinusoid. Okay, and there's a stave cell in between. Okay, so there's either a closed circulation where like the pericillar artery ends into a sinusoid, or the penicillar artery ends into any open space and it just like diffuses inside. Okay, this is it. Alhamdulillah, we are done. This is everything regarding the 3 lectures: the muscles, nerves, and the lymph, everything. If you guys have any questions at all, please do not hesitate to contact me. Here's my number and here's my Gmail. Text me on WhatsApp صراحة, I'll answer على طول. Here's the QR code if you guys wanna scan it for the the feedback. And also there's some questions inside. So if you want some practice questions, there's some inside that crash course. You're very welcome.